University, and our guest of honor, Colonel Rajesh Kumar, General Manager, NHIDCL, Government of India. I, Anshuman Dwivedi, welcome all the participants on behalf of Botanical Survey of India, Sikkim Himalayan Regional Centre, Gangtok, Sikkim, to a very special day where you all will learn and get guidance on plant identification in this capacity building training program. We are glad to work with all present. Now, I invite guests to today's function with a request to come on position and occupy the distinguished chair. So, we are going to come forward. Now, I would like to request our head of office, Dr. Rajiv Guru, to kindly speak and head of office to honor and welcome our chief guest, Professor Dr. Jyoti Prakash Saman, Professor and Head Master with Harvard. I'd like to request Dr. J. Franklin Benjamin kindly speak to honor and welcome our guest of honor, Colonel Rajesh Kumar with Harvard. I request Dr. Rajiv Guru and his team head of office come to the dais and for welcome speech and to talk on the theme of. Thank you, everyone. We are very privileged today, and we have gathered today here for a first training program in planet. This is a part of the See, I'm very thankful to Sir Dr. Jyoti Prakash Saman, who in not only my view, but it is our all of our view, who is the leader in the field of microbiology, not only in India, but this total Asia, because we is having lot this collaboration with the Japanese and other international people. Last 2 years, I'm serving here. Before that also I know him, but he did not have much interaction. So just recently I met him in a program, and when we were planning for this, and somehow it was that I got that Samantha should come to us this in Botanical Survey of India, and this is I find is a very good opportunity that because all the young people gathered here for this training program. Particularly our this main objectives of the program is that everybody is talking about that taxonomy is dying. And it is of course true. And to some extent the blame goes to the taxonomy itself. Because we are not able to make it too interesting that people will be interested to go and watch. So what is happening, many people, many students, they are more interested for doing molecular and other applied aspects. Phytochemistry, nutraceutical activities, antioxidant, all those things. But very interestingly, everybody should know and realize that if the identity is not correct, all things will be. So that's why the fundamental of plant taxonomy or identification, if somebody will be making, because I sometimes go to many meetings and I find the people who is presenting their research, the basic identity of the plant. Because just recently I will tell you in one meeting I have seen that a person was working with the phytochemistry and they have extracted a lot of molecules from a plant, which is actually Pistar clarity, but his own report and even they have published in some high impact journals as Pistar mutants. And most interestingly, in NCBI, people are putting the DNA in the name of another species which exactly is different species. So you see how fundamentally we are creating huge problem and a mess. So if the things will go like this, we will get a lot more problem. Because this is why it is happening, you know. It is happening because we are so casual in getting the identity of a plant or something. Because if we ask to a person who do not go actual to the depth how the name has attached to a particular organism. Because there's a lot of things which is related to typification, type specimen, all those kind of things. So if we do not know fundamentally all those things, then these type of problems will get they will get more and more problem, and this will not the benefit of science or for the mankind. So that's why this type of things, although Botanical Survey of India has this mandate, but this Sikkim Himalayan region, we are very new in organizing this kind of a thing. Just recently in 2022, in 3rd February, we have started one training program, and that was I think review of this that was for ecology, for quantification of the forest different species and something like that to know how we can do this. And there in that 3rd February program, 13 students joined us, and that was a success for us, and we thought that we should carry over these things. And this time we have when we have planned that we were not expecting too many students like on this way. We were thinking that only 22 students will accommodate and will do the program. But what happened? It was so much overwhelming response that about 40 students approached us for this program. And this from our headquarters has told that why we have to do only offline, because after Corona, online is also a very important thing, and that's why it has been given some advertisement in Twitter and something like that. And today, in this program, it is a standard program actually. We are here, this first session will be offline here, and second session we will be online, and the program will be on camera. They will be online for this. So altogether, 327 students has registered from all world, and this 327 students is watching us now in YouTube live stream. So out of these 85 students are from Gujarat, 55 from Maharashtra, 29 from West Bengal, 27 from Assam, 22 UP, and from almost all over India from this Andaman and Nicobar also 2 students joined. So this is a very good means sign, signal that people still wanted to know about taxonomy and this type of fundamental part of the basic science. So I am happy, and I am happy that our partner organization, because one here project is going on, which is given by NSIDL, Honorable Rajesh Kumar, he also is here. So we are considering people as our partner because when we got some funding, project is going on. So that's why I welcome you all. Particularly, Aman sir, I'm very thankful to you, sir. You have given us time and your presence definitely encouraged us and Manoj also. My very beloved students and my juniors, my colleagues, thank you that you all have come and I think you are helping us to make this program successful. Thank you very much.
Thank you so much, sir. That was really motivating and extremely informative. Now, I request our guest of honor, Colonel Rajesh Kumar, General Manager in NSIDCL, Government of India, to the dais and say a few words. Sir.
I'm thankful to you for inviting me for this lovely morning here today. And it's my honor to share a dais with him. He's a person of eminence and esteem in the state of Sikkim. I'm honored, sir. Dear participants, good morning to you. all those who are present here and probably if some of us are online from various parts of the country. Actually, I wanted to be one participant like you to get a chance to attend the program, but I am not as qualified as you people. You all are here for a capacity building program, but you must realize that you are really, by doing this, participating into a grand national assistance program. You are in a way contributing to the country by acquiring this knowledge, which certainly you will be delivering up to some time. We are not very, casual about the environment, and in fact, nobody bothers about environment nowadays. A human being, by nature is a little selfish. Everybody thinks, yes, a lot of threat to environment, we keep reading, there is a lot of media hype about it. But at the back of our mind, we always think how we are connected to this world for next 20 years, 30 years, 40 years, 100 years, 200 years. That's all. And this is just kind of nothing to it. But actually, any contribution that has to go into the environment, that has to be something, is done 200 years, 300 years beyond. And that kind of work methodology comes from this kind of science in which you people are actually going to participate, or going to be a part. So, standing here, my pray, prayer to you will be that this small little program one day of capsule, contribute your maximum and try to learn the maximum out of it. Don't leave anything to doubt. Whatever is the agenda of the program given, find it, chew it, grasp it, and take it with you. You are going to have a very expert faculty who will be guiding you on this, right? And you have the esteemed expertise from guest of honor here to make the best of it. All the best to you. Thank you.
Now, I would like to request all the participants to introduce themselves one by one. Starting name and service and name and everything.
My name is Shalini Sethi and I'm from Botanical Shalini Sethi.
No.
My name is Shalini Sethi from the Botanical College and
Myself, Anand Singh, and assistant botany, regional biology.
Morning everyone, my name is Pooja and I'm also the scholar from Sikkim. Hello everyone, my name is Mamta Sethi and I'm a research scholar from Sikkim and I'm hosting my PhD from Mizoram University. Good morning everyone, I will have to live Hello everyone, good morning. My name is Anita Sharma. I'm from the
तो दो लिया है ना, मैं देख रहा हूँ इनॉग्रेशन प्रोग्राम देख रहा हूँ।
Everyone, I'm Marcusita from study and see what. Good evening, everyone. My name is Yashapani and I'm one part of the University College.
I did.
Good morning, everyone. I'm Samantha Chetia, research scholar from the University of India. Good morning, everyone. I'm Pratima Mukti from the University of India.
Good morning, everyone. Good morning, everyone. My name is Arnab Sabha. I'm a research scholar at the Department of Zoology in University. Department of Zoology in University.
Good morning, everyone. My name is Chaman Bhutia. I'm postgraduate student from the Botanical Survey. Good morning. My name is Mohammed Lacha. I'm from NSBDC and I'm from the Department of Botany.
I'm Madhusudan Khanna, this is covered by the Department of Botany.
Louder, please. Louder.
My name is Mohammed. I'm a research scholar from the Department of Botany.
I request all the also let me get questions.
Good morning, sir. My name is Saraswati. I'm a technical assistant of this project. Yes.
Good morning, everyone. My name is Victoria. I'm from NSBDC College. Hello, everyone. I'm Nirmal Sana. I'm currently pursuing masters from the Botanical College.
How 6 semester? Okay.
Which semester? Thank you, everyone, and welcome to study. I'm Anshuman Devi, working here at LDC.
Our people also included.
Good morning, sir. Myself, Dr. Basant Kumar Singh, working as botanical assistant, and today looking after the social media telecast. Thank you, sir.
Good morning, everybody. I'm Ratan Giri, staff of Botanical Survey of India.
Very important person from my department. He is retiring. These are the people who is retiring in government of India. Actually, we are not getting back of this kind of people now. This is the biggest problem. Running this kind of institute.
I'm Dr. Chaman Bhutia.
Myself, Paul Sepa. I'm senior preservation assistant. Yes.
My name is Vijay Saha. I'm working as a library technology assistant.
Thank you very much.
That's not it. It's a non-card here. All the specimens we have seen, no? These things, BD and this. And Pasandi, first they had all this stitching and those things. Actually, specimens need to be stitched after the mounting. So it was so now. What was about
32 years.
And we would like to take the opportunity to introduce our chief guest company, Professor Dr. Julie is a senior professor in Department of Microbiology, Sikkim Central University, and is the pioneer researcher in the ethnic communities and beverages of Asia for the last 25 years, focusing on medicinal genomics, metabolomics, bioinformatics, probiotics, and health benefits. He is also holding the prestigious International Center for Integrated Mountain Development (ICIMD) Mountain Chain. He is a fellow of National Academy of Agricultural Sciences, Indian Academy of Microbiological Sciences, Biotech Research Society of India. He has received several national and international awards, including Department of Biotechnology, Biobank Award in 2005. He has published more than 180 research publications, including 8 books, 28 book chapters, with more than 6,700 citations, and H-index of 46 in scholars, and produced 16 PhD students. I request our chief guest, Dr. Professor Jyoti Prakash Kaman, to the dais and to say a few words. Big applause for her.
Namaskar. First of all, thank you very much for inviting me. Dr. Rajesh Mogai, very dynamic, renowned researchers, myself, Rajesh Kumarji. It's a pleasure for me to introduce you, and you are taking a very good, important assignment in the infrastructure development of the connectivity. And very happy to know that you have taken place on the environment. The environment is very important for any kind of development. Just to know the environment is very important. That is supposed to be plants, because to protect the and such and all. And I'm very happy that you are aware of that, and all the staff, the scientists, and the other non-teaching staff of the BSI and all the participants. The very important of this today's workshop is the orientation, is the capacity building on the taxonomical tools to identify for the identifications of the plants, which is very, very important. And I'm very thankful to the BSI. The Director General, Dr. Mao, is my very good friend, and we have been going for the last 30 years. Been trained in Kew Garden in London. Very, very eminent scientist, very, very sharp focused, and we have been working together in many occasions. Very happy that Sikkim has several national institute. Of course, Sikkim has the full-fledged Central University where I belong, and I am the founder of the Sikkim University in 2007 we started, and this is the 12th Central University in the Northeast, and we have the oldest college where I have started my career in 1986 in the Department of Botany, where most of you have not gone. And I served in Sikkim Government College, now present duties for the Nonpal Mandari Degree College. I am the founder of that department, and still the laboratory is there. when I joined the Sikkim University as a first registered in 2011, I left that and handed over to the government. And I made that at the time of the 2 crores laboratory. And I'm still happy that afterwards the government was very pleased and they actually once you leave those Sikkim government colleges affiliated to our university as well. But it is a government property. I had a project, I developed that, I developed the research environment. And when I handed over and joined the university, I handed over that to the administrator, to the principal and the head of the department. But later on, the government, I was told that the minister visited and he asked that very beautiful, of course, it is very beautiful and very established laboratory, I think what minister is doing. And the minister said that no, it should be handed over, it should be given to the Professor Kamaman. And I said no, because I am no more employee of Sikkim Government College. I am an employee of the Sikkim University. So what the government had decided that, because my wife is also working here, she's the head of the Department of Zoology. So government handed over to the head of the department, Dr. Kamaman, not to the head of the department of Botany. I think you know about appreciate that. But anyway, now we have started the Department of Microbiology because of my initiative project. Very thankful to the chief ministers. And we are trying that. And from this year, the Department of Microbiology is already here, and 2 of our students are here as assistant professors. Now I come to the, see my background, I did my BSc honors in botany in Botanical Government College in 1984, a BSc in 82, then MSc in 84, 85, 84. And as you know, that the Botanical Government College was again regarded as the second presidency college in India. So Botanical Government College, we had a very fortunate. There was, of course, the North Bengal University was there, but there was no department of botany and zoology. It was in the Gazi Government College already. So I had a privilege to get a good education in Gazi Government College. Though I have been, I got admission in the Presidency College in Calcutta, but I preferred to study in the Gazi Government College. So that time microbiology was not there. There was no microbiology, very few. Pune University and few universities only had microbiology that just started. And there was no biotechnology, we never heard biotechnology. We never heard of bioinformatics, we never heard of metagenomics, we never heard of anything. Machine learning, nothing and all. Even molecular biology was also very rare in our syllabus. But we had a basic knowledge in the botany. And we used to go for the excursion. That time, the excursions was compulsory. The collection of the plants, though it is banned now because of the Biodiversity Act. You cannot collect the plants. You can just get the digital pictures, images, only that. But that time, there was no such act. So we used to go every Sunday. We used to go to the different places. I visited almost all the villages, all the forests in Darjeeling, California, areas and all. And when I joined here as a lecturer at that time in 1986, as a young lecturer after getting my master's degree, and I just joined the PhD. And I joined in Sikkim Government College. Then there were only me and Dr. Salman. She died a few years ago. And we started that department and all. So I was young, I was not married. Then I used to take the students, the first batch students, second batch students, every Sunday to all places of in and around Sikkim. So just imagine, the 1986, I was hardly 24 or 25 years old.
I do not know about the young assistant professors of our university and our colleges, whether they go to the different places by foot. Now, I do not know. But that time, there was very limited transportations. So every Sunday, we used to go, I used to cook some food, I used to buy some the foods for the students, and they used to carry something, and we used to go right to the Bangla and everything, Rangka and all, Busuk and all, by walk. I have almost, now I cannot walk, but I walk. Sometimes I show to my children that I walk this, all this, my past, everything. Room take area and all that used to come. Because there was no entertainment, and we were all acquainted with plants. And we used to go sit in the forest or in the village with full of the plants, and we are acquainted with the beaches, Jugasana. Okay. So I have not seen the Juga for many years. I love to see Juga. And next week I'm planning to take my family to village to show to see the beach. So we had a life of everything. This is how I have been motivated and all. The plants are motivated in that way. Though my specialization is microbiology, food microbiology, and the microbiology also comes under the plant. And all the substrate, the plant-based and animal-based. But at least what was very important in taking the botany is when I was given a chance to understand the plants. That is, the plants cannot speak, the plants cannot move, but the entire sense of their, the sense and their presence, you feel so motivated and all, and you like all greenery and all. The beautiful flowers, inflorescence, different type of leaves, different type of roots, different type of states. Not only plants, not only angiosperms, the gymnosperms, the bryophytes, the beautiful bryophytes, lichens, the algae, and some fungi, mushrooms and all. So, very, that type of the motivation that was given right from my the young career stage. Now I'll come to the point that why taxonomy that Dr. Gogoi has rightly said. The taxonomy is very important. What is taxonomy? To identify any living organism. Identity is very important. There is a politics on the identity. There is a war on the identity. There is a war between the Ukraine and Russia because of the identity. Because Ukraine wants to have its own identity, though it was ruled by the Russians, but they are not Russians. So everybody in our country also the politics, the identity. So I'm not going into the politics of that. But identity is very important. Just like just a human being, just for example, me. Okay, my name. Who am I? What caste I belong? Suppose I belong to Tamil. And Tamil also there are so many subclasses, like and all. And under that also there are so many. Like Mount Abu, there are two types of law. One is Jashirban and Upal. Mount Abu, Tharu. Tharu are pretty Nepal. And there is a Bodra. So you go in that way. So when the priest is performing any puja, he wants the identity of the family, the dynasty. Every family has a father, father has a father, father has a father, mother. So that is called the identity. Same in the plant also. Same in the plant also, when you go for it looks same plant, but it has got its own identity. To give that a sign, it belongs to some family, it belongs to some kingdoms, that orders, then it belongs to genus. For example, Tamam is a genus, that is species. Suppose I belong to one, that's a species. So it also has a species. That species also, there are some Rudra, that is subspecies. And now with the molecular taxonomy, it goes to the strange, substrange and also. Identity of any organism, whether it is a plant or animal or human being, is very, very important. Dr. Gogai has rightly said, today we are working, there are many scientists working on the byproducts. Okay, they work on the antimicrobials or byproducts or some compounds and all, extracted from either from the plant or from microorganisms or from animal source and all. But ultimately, they do a lot of work on the molecular work, the bioinformatics and all advanced work, and further pattern also, and which are sold in the market also. But the important is the source, from where this has been identity of that plant. The identity of that microorganism is very important. We are working on the microbial, antimicrobial compounds, but the identity is very important. Now there are two types of identity. One is the phenotypic identity and there is a genotypic identity. And now that is the old culture. The recent culture is the culture dependent and the culture independent. The culture independent comes the phenotypic and the molecular. That means which are like the plants, which are culturable to the medium, and which are shown if the colonies are grown, then you can assume that, okay, this is, then you do for the ground study and some morphology. But scientists have said that in that particular food or particular samples, there are many unculturable microorganisms also, which cannot be seen by our eyes and which are not grown in the medium. So we ignore them. So we ignore that. So we report only those who have been seen by our eyes, but which are not seen by the things are huge. And they have got huge potentials for many biotic compounds and all. Then the new technology has come, what is called maybe eukaryotes, it may be the prokaryotes, that is bacteria, it may be the eukaryotes, it may be the fungi, it may be the virus, it may be the archaea. The latest technique which we are applying now in our lab, my lab, is succulent sequencing. Same in the plant also. The basic identification is very important. When you go to the field, any plant or no plant is given to you. And what is important is the ethnobiological knowledge of the people. The people say that this plant has got the therapeutic uses. It can cure the diarrhea, it can cure the stomach ache, or it can cure the headache. That is not a traditional practice recorded. Now, as a scientist, validation of that, whether that the traditional healers which they have said is right or wrong, whether that plants have got biofunctional properties or not. So to do that, first is you have to identify. You have to identify, you have to give the proper identification of that plant. Village, take the plant, take the different type of the leaves, and in the leaves also with the magnify, there are so many veins, the different artistic veins, each and every species of gene, the nature of God has given a particular organism, is a particular, that is called a signature, molecular signature. And that is given only to that particular orientation of the veins. the both and the color of the leaves, color of the flowers, inflorescence, roots, this and that. These are all morphological. And morphological documentation, identification is very, very important. Then after that, you have a herbarium in the PSI comparing with it. Then, once you go to the taxonomical keys, there are so many taxonomical keys. The father of the taxonomical keys is the Joseph Dr. Hooker, as you know. Everybody knows he is the father, Joseph Dr. Hooker. But do you know that who was Joseph Hooker? Joseph Dr. Hooker is also the founder of the famous Darjeeling. It was Joseph Dr. Hooker has not discovered Darjeeling, today there was no Darjeeling. It was full of forest. When he in Sikkim, Gangtok, then he made that one. There was a war between the Prithvinarayan Shah, where he was going for the unification of Nepal, all these territories, the West, Sikkim, and all. And he wanted to make one country, Nepal. And he came up to Sikkim and he converted Darjeeling also. At that time, Darjeeling was a part of Sikkim. And 1816, the Suhori Treaty was there. And the British already came, their headquarters in Calcutta, not in Delhi, Viceroy was there. So, then they handed over to the Chogyam, Darjeeling. Darjeeling was not the present Darjeeling. Nothing. It was just a forest, a small Gundri Bazaar, a few villages and all, nothing was there. So, and then 1819, roundabout, Joseph Dr. Hooker came from the Cube and he wanted to explore the Himalayas, the flora. And he came because he is a very eminent botanist, very decorated botanist. He was given the full the support from the British government. Campbell, that time he was a superintendent, and he climbed that Hillcart Road because it was a dark road. If you have traveled that Pankhabari road, that was not this big road. That was a small road. He traveled for at least a week, 10 days to reach Darjeeling. Actually, his intention was to go to the Rinchaipur, the West Sikkim, to explore the Himalayas, mostly the rhododendron, and he has collected all the matters. He wanted to go there, and he took the help of the local people, most of the Lepchas, the Hindus, some Pahans, or whoever were there in that areas, and he made Darjeeling as a base camp. So they reached Darjeeling. Moment they reached Darjeeling, they made a thing. It was a marvelous, magnificent view of the Kanchenjunga. And both Campbell and Sir Joseph Dr. were very impressed. And they have never, and they compared it, I mean, the weather of Darjeeling was almost same as in Switzerland. Scotland, in London. In Scotland. So, then he went to the Rinchaipur with the help of the Lepchas and he planned and all. Then he was arrested also by Chobel, that there was some dispute and all. Then after that, then he came to Calcutta. Then on the way to the queue, but he submitted a report to the Viceroy. He didn't submit his findings because he had to submit to the queue garden in London. But he submitted that, Viceroy, I have seen Darjeeling is a very beautiful place, and you please make the summer capital of India in Darjeeling. Okay, that immediately, 2029, the Viceroy ordered, then they went to Darjeeling and they found that they started constructing all the lane and all the roads and all by 1836, the beautiful Darjeeling came. This is the history you should know as a politics. It's always linked. Hooker's book is always linked. If you want to read it, these books are there in our central library. great it is. They have the written every, and not only that, he has written, I've read that the people of these regions drink a chant, the points of the chant. Bodhogojara has mentioned there, people, and he has mentioned some ethnic foods also, which he has taken out from that. So this is how the Joseph Darton is not a botanist. He has been regarded as the founder of the Darjeeling. And after that, the Britishers made it, and the railways came, tea gardens came, tea garden came in 1849, and the railway came in 1879. And the first hydro project in the Asia, Chitrabong, came in 1894. I know. एउटा गीत छ नि के अरे कम्पनी सर्भ यता सिक्किमपट्टि धिब्रीमा हुन्थ्यो अरे तर कम्पनी सर्भ चाहिँ एउटा कम्पनी भन्छ टी गार्डनलाई कम्पनी भन्छ we used to call the company now also older people say that company सर्भ टी गार्डनमा चाहिँ झलझलको बत्ती बल्थ्यो उता पारि सिटीमा चाहिँ धिब्री बल्थ्यो and the richest municipality of Darjeeling at that time. Now, what is Darjeeling Himalayan? So anyway, that is the history of, so as a botanist, then once you identify the plants and all, morphologically, phenotypically, confirmations by the molecule is very important. Without molecular confirmations, no taxons or any identity is accepted by the international organizations. It has to be identified by the molecular tools. So there are so many molecular tools. The taxonomy of in the plants also, in the animals also, in the microorganisms. Now, the latest technique which we apply in the microorganism is, I told you, the shotgun sequencing. With the help of the sequencing, and it has to be decoded with the sequence. You get only the sequence, the sequence vision, which has to be readable form, and that readable form has to be decoded by the techniques which is called the machine learning, that is the bioinformatics. And that taxonomy is called the meta-taxonomy. So in the plants also, it has been done that in the plant also, you take a piece of plant and apply the metagenomics. I think nobody has done, you can do it, certain sequence, and you can see the entire everything. You don't have to do anything, everything plant, and we do in the treatment. So we have not tried in the plant, but if you want to see our level in our department or our departments, we have standardized that certain sequencing and all. Another important is the plants, not only taxonomy. Applicability is very, very important. Do you know, because of the COVID, the plants have many secondary metabolites. What are metabolites? Nowadays, the food value, protein, carbohydrate, fat, these concepts are gone now. Now we go in the bioactive compound. The food which can boost your immunity, the food which can control your obesity, the food which can control your diabetics, the food which can give you the glazing thing is all in the plant. What is that? That is the metabolites. There are two types of metabolites. One is secondary metabolite, one is primary metabolites. Primary metabolites are mostly amino acid, fatty acid, this and that. But the secondary metabolites are small biomolecules. So which can enter into any part of the body, any compartment of the cell, because cell has to be different. There's a cell wall, very thick cell wall. So all molecules cannot enter. But this metabolites, the secondary metabolites, which are called the untargeted metabolites. This can go into any compartment of the cell and they enhance all the immunity, all the biotic compounds. And who are they? This non-targeted metabolites are immunomodulators, the microvitamins, okay, bioactive compound, isoflavones, which are got a very high demand now. And there is a technique that is called the metabolomics, and which is applied in the plants also now. Mostly in the drugs, it is now the latest science, and we in the microbiology, food microbiology, we have already published several papers also. We have also found many, for example, we have been working on the fermented soybean product that is commonly called kinema in Sikkim, Darjeeling, Awaja in Manipur, Ekam in Mizoram, Memarthu in Assam, Assam ka area of India. All these are the plant-based. In that plant, which is the soybean is very rich in the metabolites, and mostly the untargeted metabolites we found. And what we have found is, after doing all the data taxonomy, each and every identification of the microorganism, and we did the validations that each and every microorganism, what is their role in the production of the metabolites. So we found that the subtalis can produce a very good secondary metabolites, mostly the untargeted metabolites, which can use to boost the immunity. So these are the immunity boosters is very important in terms of the COVID. COVID is caused by the virus, the smallest organism in the world, and the deadliest organism in the world. I should go to only male, I should not go to female. I should go to only Tamil, I should not go to Lecha. I should not go to the secretary, I should go to the pure. No. I should not go to the chief ministers, I should go to the farmer. No. It will go to everyone. Is it there? But everyone will not get that treatment. Why? Because everybody has the immunity system. And why immunity system is very important is everybody has an immunity system. And those our organisms, you are working on the plant, but think about that. Every organism is full of every living organism is full of microorganisms. हमारे पास जो है हमारे शरीर से नीचे तक all the microorganisms, and they are all friendly, right from brain to our throat, to stomach, and all. If there is any disturbance in this organism, that is microbial, it is called a microbial, then there is a headache, there is a stomach ache, there is a cancer, so many other things. So who will repair this? Organisms which are outside, and these are mostly present in the fermented food, mostly in the probiotics, that is called the probiotics. They go inside, if you eat, if you take the heat. Actually, you are not taking the heat. I think you have seen the heat. And that will go, if you see the contrast microscope, you will see the moving organism, that is lactobacillus. Actually, you are eating the microorganisms. When you are eating enema, you are actually eating the microorganisms. These are friendly, these are not foreign. And they repair our belonging to the meta taxonomy, and we do the reverse. We do the first circle sequencing, get the samples and all, and we get all the bioinformatics, functionality, we get the entire pathways, get a meta taxonomy, all that is meta right from the families, all everything and all. Then we find the abundant hypothesis, more than one person. Whoever is a dominant is, and we can get the link to each and every metabolic activities, metabolisms, hospital, untargeted metabolic vitamins and all. Then we select that, and now culturally, we get a special media to grow that, and we prepare this. That is a reverse. Initially, the scientists used to do the cultural dip at that, and then after cultural dip. Now it's a reverse. We do. First, cultural dip, we get the entire background. Then we select only those who are unwanted, we don't select, only select and do special media and do other tests and all. That is the cultural dip. Now, my dear students, most of you are PhD students and all. And yes, I is doing very good work. Because see, taxonomy is nowadays it is very difficult whenever we appoint an associate professors in department of botany or in geology, we hardly find a classical taxonomist and all. But it is in syllabus we have to do. I do not know in many colleges and universities. There are very few. In our times, there were many taxonomists. But nowadays, the taxonomy has mostly switched over to the biochemistry, this and that. Okay, no problem. I'm also a taxonomist. I'm a microbiologist taxonomist. But we do from the metataxonomics to the molecular. That's what I say that whoever are doing the secondary metabolites or compounds or biochemistry and all should have a very strong knowledge of the taxonomy. Without the identity, you cannot do it. Now, my last message to all the PhD students is nowadays, how many papers you have published that will not count? I have published 100 papers, I have published 10 papers, but what is your impact factor? And what is your citation is very. You publish only one paper in India, or in your PhD only 2 or 3 papers, but published in a very high impact factor, more than 3 impact factor. That will count. And whenever you go to any interview, they will say that, for example, in my department, and I have told all the teachers, all the faculty members in our Sikkim University also, I'm telling them research is the first, and every student, whoever is in our university, to get into the PhD is very difficult. We have a rigorous selection procedures, and whoever are selected in our PhD programs are the best of the best from India. And we train them. And very important thing is that when you finish your PhDs, when you submit your PhDs all, whether I from BSI or the GV or thing, be sure that your cumulative impact factor should be 10, not less than 10. You make a target like that. So in my department, in my lab, 10 is the basic, but most of my students get more than 20 years also. You work hard and get always the new. Do not replicate the others' work. Do not just do the stereotypical work. Always find the research gap. Where is the research gap? Who has not done that? Always do some new technology. That's why your paper, that's why your citation will go very high. Everybody will refer your paper. That's why the journals, now it's very difficult to publish in journals. Best of the best journals, even after paying also. All top journals are paid also, but they give the web also. See your quality of the paper. The novelty is very important. Every journal puts a highlight or the graphic abstract, and from the editorial officially, your papers are rejected. Rejection rate is very high. And whenever you do your research, one is experiment clear for what I am going to do. More clear or not, you should know that also. And your hypothesis, the God has given the thinking power. The artificial media can do that, whatever we can do that. Predicting, that is a predictions. I'm going to do this, with this result, I'm going to find this, and I'm going to publish this. You make a whole protocol. Then you work. What you as a student, you have to learn is good, meticulous working, and you generate so many data, and many students do not know how to compare. When your supervisor says, "Abhi paper start karo, abhi mujhe graph bana karo," it takes 1 year for you. So learning that is also a technique. How to compile that, how to use the different statistics, how to use bioinformatics, and how to compress your data into a tabular form, or a graphic form, or what is called readable form. Then after that, your writing skill is very important. Many students do not have the good writing skill. If you do not have the good writing skill, if you cannot make your own phrases, there is a one term that is plagiarism. Now university uses it. The PhD thesis should have more than less than 10 plagiarism rate. Which are tested. Because if you copy here and there, copy and paste, your plagiarism high. And there is it is a crime. It's a rule. So how to reduce that plagiarism is make your own English, making all your phrase. So how to make your phrase? You have to be very strong in English. You have to write, you have to start the writing and speaking. If you feel shy to speak to others, you speak somewhere then Zuluko Terry would like it, but the other unknown. So they develop like a Then after that, very important is presentation, orientation. Your orientation should be, you have done a good work, very good work. And when you are asked to make a presentation, this botanical, the syllabus, then people will just okay. So in practice, that is why in our university, every 6 months we make our presentations. And every time we make a presentation at all. So presentations is very important, not only for your PhD, when you apply for the assistant professor, when you apply for the scientist, that introduce the people. We'll see first your personality. What is your personality? I'll give it a secrecy. Think that I am taking your interview. Is first is I do. Mero akhir then is the label of so you are not selection committee will think that you are not a good teacher. Whatever good publication you have, whatever they will answer. Teachers should be, a scientist should be, a very, very progressive, very, very, the communicating is very important. Okay, so when we select the assistant professors, any university, especially in central or any scientist, is your internal personality and he has got a very good communicative skill that is very important. Very good. So that will always make a good success. So this is my gifts to you, and I think I have already been telling to my students from Sikkim University, those who are from the Southern College and the USI and Sikkim and other institutions, Science School. Please learn this. And another is, please come to our university. Our central library is open to all. It is not only for the students of Sikkim University, it is for all. We have got a very rich, all type of the e-journals, all types of access, everything is there. Only the thing is that there is certain rules that you have to register or you have to get some letter from your institution, doesn't matter. So you can access that with each able. And another is, I'm very thankful to Dr. Gogai. This is a good platform that you make a mixture of all the researchers working in one platform. This is very important. I see in our university only students of botany will go and hardly some in zoology, and we don't mix with the students of government college, or we hardly meet the students of the BSI and office. Okay, and we could have invited the students from micro as well. Okay. So this is what the thing in orientation, you should know each other and share your mobiles and your WhatsApp and all. Of course, you share in many things, but this is the very thing. Because you use your mobile in a very constructive way. Your mobile is a dictionary. You check for so many Amazons, shoppings and pictures, some other chattings and other things. But you hardly use your Google for identification. You do it. All the systems are there. All this data taxonomy, every NCBI, all data accessible in the Google browser. You do that, then you have it. So once again, I think if you have any questions, anything, and always do PhD is very important. In India, we have to do or promote our higher education, and the science is very, very important. And you are now going for the what are we doing? We are not only doing the PhDs, not only doing the artificial plants, the animals, the microorganisms, but you are doing national building. This national building is very, very important. Our country needs scientists like you, and we are very sincere and dedicated scientists. All the best.
Thank you, sir. We are so honored and privileged to have you among us. I request Dr. J.H. Benjamin Franklin Scientist to felicitate our Chief Guest, Professor Dr. Jyoti Prakash Aman with short. A big round of applause. Professor Dr. Jyoti Prakash Aman with Solo Sikkim.
Important thing that ignited with this war, the young generation. You didn't know. And in front of you, you people are seeing this because this is that where the Rotan and Datan Hokari is living the place. And I know this is not only for Sikkim or Darjeeling or something, what Sir has told us that Darjeeling was almost a lost city, which has been discovered by Mr. Datan Hokari. This is not only for Darjeeling. If Joseph Dalton Hooker would not have come to Darjeeling and Sikkim in 1848, the whole world's history of botany would have been different. Whole world's history of botany would have been different. And that botany would have been not written from the land of India, the land of Darjeeling, than from the land of Sikkim. This is that what we all have to realize. This is the thing somewhere I find that the new generation is somewhere is got lost or something that we have to believe. See, when he came in 1848 to Darjeeling, he was simply a 24 years old boy. Maybe he is younger than many of you people, but he has written a chapter in the history of mankind. It is not the history of science, but it is in the history of mankind. That what a scientist should do for with the intervention of science to make the humanity go forward. And that chapter has been initiated from the land of Darjeeling and Sikkim. This is the history we should not forget. And that's why, because of Joseph Dalton Hooker, the whole world is not only the science of botany, but the history of science, history of mankind has been very differently been taken care of. And if Joseph Delgamuta would not have come in 1848 to Darjeeling, today we would not have sitting in this hall, and we would not have made this meeting, because the whole thing would have been very different. So, sir, in 2019, I joined this department as 1979. I think with the instruction of that time our director, Dr. This Sikkim Himalayan Regional Centre has been initiated, and the man behind this is BTN. That is also your friend. So till then, we have tirelessly working to document the floral wealth of Sikkim. And when I in 2019 I joined this department, my present director, Dr. A.M. who already we have told, he was a PhD student from Reading University and Royal Botanic Garden, Kew. And he told that because we are going to Sikkim, the data which has been generated in the last 40 years that has to be brought as well. Because that is the aspiration not only from the people of Sikkim or from the Sikkim Himalayan region, but this is the aspiration from the whole country, because Sikkim is the most diverse state in terms of biodiversity, because here, although it is geographically less than 1%, but Sikkim we contributed 26% of the biodiversity for the country. And in the whole Himalaya, it is from Afghanistan to Vietnam in China, 48% of the biodiversity is contributed. So, sir, taking the pain with all other earlier data and all my colleagues, so we have brought out this. So we are very happy with this, our gathering and our talking.
Now I request Mr. Nurbu Sherpa to felicitate our chief guest, Professor Jyoti Prakash Sharma, with white balsam. Now I request Mr. Ratan Giri to felicitate our chief guest, Professor Dr. Jyoti Prakash Sharma, with movement. I request Mr. Mihi Sahab to felicitate our chief guest, Professor Dr. Jyoti Prakash Sharma. I request Now I request everyone to come to outside of the office for a group photo.
session of this one-day capacity building training program. So here we are going to talk about basics of taxonomy, especially the identification part, or why or what you have to do before identification. So everything needs an effort. So even for taxonomic identification also, you need some kind of an effort so that it will be easier for you to identify the plant. I can't promise you that in one day or this one session you will be learning everything and able to by evening going and identify all the plant in Gangtok. I don't say, I won't promise that, because taxonomy is more than science. It is art. It needs a lot of experience. A lot of experience. Even after 20 years, I'm very skeptical about immediately identifying a plant. I'm always very cautious because it is nature, and nature has kind of variations that you won't be able to imagine. So that with this background, we will go to the presentation. Next slide. Next. Yeah. As usual, this is mandatory. We have to introduce our institution to the audience. Before that, before we go to our technical session, you all know BSI was founded in 1890, and Sir George King was our first ex-officio director of BSI, and the lady was the instrumental in bringing BSI back to life after independence. Her name is Janaki Amal, and she was the first ex-officio director of BSI, and she only reorganized BSI and organized the BSI into what we see today. Okay, next. So we have so many centers all over India. These are all called regional centers. So SHRC Gangtok, Sikkim Himalayan Regional Centre Gangtok was established in December 1975. Next. And other units of BSI equally important is AJC, Indian Botanic Garden, Howrah. It's very famous all over the world. And Central National Herbarium, of course, one of the biggest herbarium in the world. And Central Botanical Laboratory, Industrial Section, and Botanic Garden of Indian Republic. Next. And our mandate is survey, inventarization, documentation, and conservation. And our primary objectives are these. Next. And our secondary objective, one of the secondary objective for this is capacity building in plant taxonomy. So that's what we are doing today. Next. And our heart and our soul, our everything of BSI is our herbarium. So we have the Central National Herbarium in Calcutta, which is one of the largest herbarium in the world, and it has more than 300 years of collection history. Okay, so SHRC herbarium The third one is when you want to identify. Saying this is the that type. Everything now the edifice that you are building. Okay, right? So this is one classic example. These two plants locally are known as Ashoka. Everybody knows I think. Okay, and one is Polyantha longifloria belonging to Anonaceae, completely different. Another is Saraka Ashoka, which is Cisalpines. This Ashoka tree has a own this thing. What was the? It is called in Sanskrit Ashoka. That means this wipes out sorrow. The Ashoka means it wipes out sorrow. That is in Sanskrit. Okay. So probably which plant? One of the plant is where in Ramayana Sita Ma sat. That is why it is called Ashoka. It removes the sorrow of the people sitting there. Which of this plant would have been done? But both are called Ashoka. No idea. This is where taxonomy comes. This is where locally both are known as taxonomy as Ashoka. And when the plant that is historically in Ramayana said as Ashoka is this one, Sarapa Ashoka. Okay, and this is a false Ashoka. This we call as a false Ashoka. Okay, this Sarapa Ashoka has a medicinal property, highly medicinal property. Your entire product will be collapsed. But fortunately here, oh, ah, fortunately here, both Centrala Asiatica and Bacopa monari, both are called from me, and both are need to know all these things only, we need taxonomy. Or else recent sciences are there, those sciences that were with the taxonomy which came into being, that also has gone back, but taxonomy continues for past 300 years. Past 300 years, the taxonomy is completely non-stop, it is continuing. So there is data for more than 1750. 1750, we take it as a starting point. Okay, that is when the Linnaeus Species Plantarum was published. From there till today, this continuous of literature, continuous piece of this thing. So the science is as new as it is old and historical. But when you ask anybody in between what a proper definition for a species, like you give for Newton's law of motion, you have three beautiful laws. There's so succinctly given, and there is no ambiguity as to what it is. But here, unfortunately, it has been discussed to death. It has been symposium, a lot of symposiums, workshops, and a lot of people, experts, everybody has discussed. But nobody could, so far, give a proper definition for a species which can encompass all the characters of a species. That is the beauty of taxonomy. That is the beauty. Because why? Species or whatever animal or whatever this thing, nature, its variation is beyond your imagination. You will say this is a species today. You will have variations, wide variety of variations. That's why as a taxonomist, to understand what a species is very difficult. You can understand a species is not a it may be a discrete unit benefit, but it is not a discrete. It is not slab. It is not 1 centimeter. 1 centimeter means you know that it is 10 millimeters. Okay, it is not species. Is not like that. In one place it will be bigger variations will be there. One place it will be smaller variations. You have to encompass all this variation. Okay, then only you know what a species is. Next, oh the next. So there are different concepts of species. There is no proper one definition, but different concepts of species. Only the first one is nominalistic, which is the local name wala. We said no. So that Ashoka local name wala, this thing is a nominalistic species. Okay, where that is given by local people for just to identify the plant by name. That's all. Okay, next is typological. It's based on the type of the organism that is at higher level that we won't discuss today. Biological and phylogenetic. The biological species only we are going to talk. We are going to talk about what is a biological species. There are different, these only 4 I have given, but another 15 are there. Different concepts. There is ecological concept of species. There is another concept of species. So many. Okay. So we are going to talk about only the biological species concept. Whenever we are talking as a taxonomist that we are talking about species, it means it is a biological species concept. Okay. Next. Yeah. So these are all the features of biological species. This is the feature. That's you have to, if you can fix it in your mind, then you will have little bit clarity about species. But this is also not entirely correct. I cannot guarantee you that this is a correct definition for a species biologically, but this is the closest I have. This is the closest definition of a biological species that I have. Okay. The first thing is this species is a group of organisms which is reproductively isolated. Okay. Reproductively isolated. Cow breeds with cow. Horses breeds with horses. So there is intermingling, interbreeding is not there, it is there. But I am not talking about that. I am talking about a group of organisms that exclusively reproduce among themselves and they won't reproduce with other animals. For example, a cow won't breed with a giraffe. That's all. That is reproduction isolation. That wall, invisible wall, is what the species is given as a definition. Next one is interbreeding. I said cow will breed with only cow. Okay, that is interbreeding. Then natural population. It should not be artificial or hybrid or lab-raised population. It is a natural population. It should be found in the nature where the species is reproductively isolated and interbreeding. And then it should have, or possibly, it should have some ecological and habitat isolation. This plant or species will thrive only in this area. For example, a camel thrives only in a desert area. It's an ecological or habitat isolation. Okay. Next is seasonal isolation. This is mostly for birds and those which are seasonal matings, like birds and other butterflies and hybrid sterility. This one is very good for animals. Hybrid sterility is very good for animal. That means if a X genus and Y genus mate, this F1 first generation will be sterile. This is the nature's, especially animal populations, first rule or kind of a thing. But even then, you have a mule, which is a hybrid between a donkey and a horse. Mule is much stronger, but it's not so beautiful. But it is much stronger than either this thing. But mule won't reproduce. But Do you think that happens in plants? Do you think? Do you think that happens in plants? Will plants F1 will be sterile? No. Sometimes it will be, mostly it is not. Okay. Lot of things or lot of, if you see a lot of food products, food are all hybrid only. But somehow we are able to make it to do F1 also, if fertile. Next. So this is the concise definition of a species, biological concept of species that I could get. That is, a species is a group of organisms interbreeding natural population and that are reproductively isolated from other such groups. That's all. So when you are talking about biological species population, you should only think of two things. It is a natural population which is reproductively isolated and there is no interbreeding. That's all. These two concepts, you keep it in your mind. Okay, next. So this is what I have to. Okay. So why that hybrid sterility will happen? That is a point number two. Isolating mechanisms that I am talking. The hybrid sterility will happen when there is a barriers to gene exchange, either at the level of pre-zygotic or post-zygotic. So pre-zygotic is your ecological habitat isolation. Okay. And gametic isolation. Next is post-zygotic is the production of sterile hybrids. Okay. A desert animal or a plant, we cannot easily hybridize with a animal or plant that is in the mountains. Okay, that is called ecological, what do you call, isolation. And gamete isolation is also when the gametes, the pollen and the ovule won't fertilize. They are not compatible. Okay, so and post-zygotic is even when within all these barriers they fertilize, male and female gamete fertilize, and after that also nature has a checkpoint in being the hybrid being a sterile. The F1 hybrid will be a sterile. These are all pre and post-zygotic barriers. Next. So, so speciation. Thus, when species, it doesn't appear like immediately, like in a flash, or you just make some combinations and put it. It becomes a species. No, it is a gradual process. Sometimes certain mutation they will be having, but usually it is a gradual population when they diverge from the ancestor population to a ecotype. Ecotype is a group of species that will adapt to a particular ecological condition. Okay, and that ecological condition, with that ecological condition, it becomes to next step, goes to next step of becoming more specialized and becoming arising into a new species. Okay, so this is how it the speciation forms from the ancestral population. Next. Okay, then there are two things for speciation which helps in the species. One is, as I told about, geographical barriers, and second is hybridization. So hybridization again comes with some caveats called homoploid speciation and polyploid speciation. I thought you are talking taxonomy, sir, and you are talking all this genetics and all. I don't understand. But if you don't understand all these things, trying to identify a species is meaningless. Just knowing a name of the species is meaningless. As much as I know, she is that, he is that, but I don't know behind what is that. That is as meaningless without understanding what the entire concept of species is there. Just naming and identifying, I don't think is worthwhile. You should know that what background the plant comes from. Next. So this is the way the geographical isolation happens. I mean, the speciation happens in the case of geographical isolation. So this is the most common way of speciation. So here, if you can see, I've taken the image from the internet. A group of insects is like, for example, there's a big flood, and that habitat is divided into. Over a period of time, the species of habitats sprout variations. So when it starts sprout variation, if that variation is favorable, that group of population will fix that variation, and that variation will go on keeping on given repeatedly. Then that becomes at one point of time a separate species. Okay, this is where the speciation happens because of geographical barriers. Next one. important cause of speciation in plants. Why speciation in plants is hybridization is one of the important causes of speciation in plants. Why? Homoploid hybrids. Homo means same. Ploidy means, what is a ploidy? A ploidy is your set of chromosomes. How much you have? Okay. So this homoploid hybrids is the origin of hybrids without change in the chromosome number. Okay. A rare form of it's a very rare form of speciation, and it is usually found in bacteria and other archaea. Archaea and bacteria are different. I hope you understand that. Archaea and bacteria, these homoploid hybrids are there. Hybrid means usually you'll have a polyploid. But with what do you call this homoploid hybrids? It's very rare in plants also. There are some senecious, some Asteraceae members. I think some other mostly Asteraceae members will have this homoploid hybrids. Okay, next one. Polyploid. This wait. This is the major event of speciation in plants. Okay. Here, because plants can show have more than a diploid set of chromosomes. Okay. It is not so in other this thing, but plants, because of their nature, they have polyploidy. This polyploidiness. This polyploidy is the extra chromosome sets. They can take, if it is possible, as much as chromosomes as possible. So this chromosome, not one single, the entire set of a chromosome it takes. This entire set of chromosome, it won't sit silently. It won't sit silently. Once the plant or hybrid has taken the extra set of chromosome, what will you do? You become Iron Man, or you become you get extra energy or extra this thing. What you do? You become different than yourself. That's what happens. Very simple. Next. It becomes the major evolutionary force in speciation. Okay, this is how speciation happens in plants. There are a lot of polyploidy in plants that helps in adapting to nature, having new set of characters, drought tolerance, stress tolerance, altitude tolerance, snow tolerance, everything. Because of that, it adapts very fastly to the environment, changing climate. Because of that, it sprouts out new species. Okay, next one. Again, this polyploids is again differentiated into autopolyploids and allopolyploids. Again, autopolyploid is nothing. Auto means self. Okay, so auto is one set of chromosome by mutation, by some aberration, it becomes multiplied. Okay, that is autopolyploid, and that is also having very good advantage. That one set of chromosome of a single plant by mutation or a chromosomal aberration or whatever events, it gets multiplied. Multiplied. So that this also helps on speciation, and it is cross within the species, whereas allopolyploid is a cross within the genera, between two genera. Okay, this is the major. Again, I'm saying this is the allopolyploid is the major diversification of species. Okay, next. So if you see, there was a review paper in which it was said that 70% of angiosperms are polyploid. That's why when you have bryophytes, pterophytes, gymnosperm, angiosperm, that angiosperms has that kind of variety, because most of the angiosperms are allopolyploids, not only polyploidy, allopolyploidy, ability to receive different sets of genomes from different kind of groups. That helps in itself to adapt itself to different habitats. That's why it is wherever you go, you find angiosperms more than gymnosperms or pterophytes or other groups. Okay, next. Now we're coming to the topic of the day, where what is identification, what you do to identify an unknown taxon. So identifying an unknown taxon, it's just a puzzle work only. So this detective work is we have to seek a pattern. What is a pattern? You have to seek a pattern. What kind of plants is what is this? It's a herb. It's a tree. What kind of this thing? What are this thing? What kind of flowers it does? What kind of where it is present? how it is present, what is its flowering time, what is its fruiting time, what are the necessary, what are the requirements of this plant. So this kind of patterns you have to see. So when you're seeing that patterns, it is the taxonomy. At one point, you have to be very close when you're coming to the species level. That knowledge you have to be very close. But at a very larger level, if you want to understand the genus, if you want to see what family it is, you have to look at a larger perspective. There are one is larger perspective, one is a smaller perspective. Both you have to see. Okay. So next, before attempting identify plants, your responsibility includes to know whatever I'm going to say. That you should know thoroughly. That is your responsibility. If you don't know this, don't try to attempt to identify a plant. Okay. As long as you know the alphabets of ABC, then you can make your English, you can read English, you can make sentence, you can talk. But if you don't know the alphabets, don't blame on English language. Right? Right? Okay. These are the alphabet. You should know the taxonomic hierarchy. What is a genus? What is a species? What is a subspecies? You have to mug up. There is no other go. How did you learn your alphabets? You mugged up. You had a very big understanding for A. No. It was said it as a A, it was A, you believed it, you mugged it up. That's all. As of now, you mugged this up. Okay. This is where it comes, what comes where. What is the division? What is the class? What is an order? What is a family? Where it comes. You have something called section and subsection. This I didn't know till even after I completed the PhD. Okay. Oh, so after that and all it will go. So you know the basics, the what I have highlighted, at least what is a species, genus, what is a family, what is an order, what is a division. Okay, division you can say angiosperms or something else. Okay, next. And next, you have to be thorough with your taxonomic characters. Taxonomic characters, you have to be thorough. If you are not thorough, then there is no use in identifying a plant. The taxonomic character is a feature of a plant. Like for example, in a plant you have a leaf, and character state of that leaf is whether it is simple leaf or compound leaf. If it is a oval leaf or it is a linear leaf, it has a parallel venation or a reticulate venation. This one all you should know. Okay, next. And you should know that obviously when you're talking about morphological taxonomy, we are very desperately dependent on the flower. We don't have any other thing. No, we are desperately dependent on the flower to identify a plant. Of course, at family level it's okay, but at the species level, we desperately need a flower to identify. So you should know what a flower is, what are the parts of a flower. You have studied it in middle school, probably lower than that. And you see, sir, now you are asking me to remember this. Of course, we are asking you to remember even the alphabets so that you can read English. So these are all alphabets, you should know this. You cannot take it for granted. Okay, next one. These are all next things that you should know. These are what, for example, what are the tree types? What are the habit types? How it is running, whether if it is creeping, how it is creeping. It is a procumbent herb or a stoloniferous herb. If it has an underground part, if it is a culm, it is a rhizome, it is a bulb, that you should know. That you should bug up. There's no another couple of slides that I am going to show are very, very important, and you have to mug it up. It is not, nobody can teach you. You have to mug it up. Okay. This is about stem and root types, leaf types. What is a leaf? What are the leaf types? Simple leaf, compound leaf, pinnate leaf, odd pinnate, even pinnate. Everything you should know. Then only it will make you sense to identify a plant. Okay, next. Then leaf shapes. Again, what are the shapes of the leaves? What are these things? Okay, and what are the leaf apex, bases? Everything where it is attached. This and all, as simple as taxonomy is, you should have wide memory power. Important thing to succeed in taxonomy is to have very good memory power and the ability to recall that when it demands. If you have good memory, but you don't recall it, then it's tough for a taxonomist. You should be able to recall also. Okay, next. So you see the surface pattern, I don't think many of you would have known. In the leaf, if there is what kind of hairs, if you look under the microscope, the hairs will be different. All this. There will be a Frocosia, there will be Stellators, there will be Villosas. Okay. With Stellators itself, we can tell which families will have the Stellators. Okay. We will know from the Stellators, if I see the microscope with Stellators, we know which are all family will have the Stellators, and we'll go directly to that, and then again see which genus it comes. So this mental mapping is very takes time. Obviously, it takes time. Okay. Next. Then floral morphology, very important. You have to know what is a flower, what kind of a flower is there, what are the inflorescences are there, what is a racemose inflorescence, what is cymose inflorescence, what is special type of inflorescence, what are the variations. Okay, next. See the corolla types, what are the, again, flower types and fruit types also. And based on the fruits itself, a lot of families can be identified. For example, legumes or the legume fruits will be legumes only, all your beans or legumes only. The moment you see, you can say this is from Fabaceae or Leguminous like that. Okay. Next, the most underlooked characters in this thing is bark of a tree. The bark of a tree has very good information. If you see, if in a lot of trees, the bark itself you can arrive at the family at the genus level. Okay. So these are all the bark of the trees which have different kind of barks, and you see that itself we can see what are the barks are there. With this bark itself, you need not even see the tree. You can arrive at the seeing the bark itself. You can arrive at the family. Next, and again there see one this last left side top corner you have seen that. Seeing that itself, we will say what is the species, because this is a unique this thing of having cauliflory. Cauliflory means having the flowers in the stem itself. That flowers have come, and after that flowers have fertilized and become fruits. The stem itself it has that is called Bakuria, and in our place it is Bakuria cutalensis. You have another Bakuria here. Okay, that belongs to four bases. We can see from that itself. I need not even see the plant there, leaves there. And the next one is Actinodaphnum bodyline. This is endemic to Western Ghats. So we can see from the bark itself. And here, the nature left was Mystica, Nima Ataluta Mystica. Based on the what do you call the arrangement of the branches itself, I can tell. So these are all, and here it's Hopia racophilia. I now with this I've seen this is again on Hopia plant. Based on this bark alone, we can differentiate between species of the genus. Okay. Okay, this bark is only for racophilia. So like that, each character will be having. So you get into the experience. Next, and for this, I recommend this book. You have it in the library. You can get in the net also. You take it and mug it up, or at least you use it as a reference. Okay. A very good explanation for everything that I have told you so far. Okay. Next. Once you got a plant and you want to identify, you have to do the literature search. First, you have to describe the plant. First, once you have read all the glossaries, all the technical terms you know, then you have to describe the plant based on the material that you have. So you will say it is a herb, it is having leaves of what, simple leaves in it, it has a raceme inflorescence, the flower is a yellow color, calyx is 4 numbers, corolla is like this, you are beating. Once that is done, after description is done, preliminarily you have to go for this kind of library in internet research. Taxonomy, as much as it is important to have knowledge in the field, equally important is to have the knowledge in the bibliography, in the literature. If either of it fails, your taxonomic judgment. Okay. So these are all just preliminary. Just if you are just immediately wanted to see some plant, refer it without any big taxonomic these things. Okay. This you go to this, set these all these things. You see if the plant matches, then come back to your definition. Your description. Again, see go next. Again, go to higher level of search. Okay. Once you search the basic that you know this is the genus or something, you go to the next level, which is very specific. If you are asking for orchids or in general flora, you go to the next specific, higher specific literature. Next. And no, you are not at all satisfied with this thing. I know this is rhododendron, but I don't know what species. I am not satisfied with the previous publication. Then go to the special. They are called taxonomic revisions. So the taxonomic revisions will be about small group, maybe a genus or a family of a particular area. Okay, a small group, and it will have most of the details, 99% of the details. For example, if I want to know what all the rhododendrons are here in Northeast India, I know this is a rhododendron, then I go to this book. Okay, next. Next year. Then, even then, after doing the revisionary works also, even then you are not able to identify. Then our last and hope is monographs. Monographs is, it will be talking only about one genus, but it will be talking everything about it under the sun. That is the monograph. Pharmacy, uses, ethnobotany, traditional knowledge, DNA, whatever it is of that genus will be in the monograph. So this is the final point. If after getting this also, you were not able to satisfy yourself, probably it is a new species. Then only you go for new species. Then, even then, you have to consult the most important thing of everything, the herbarium. Because what you study in literature and what you see in plant, based on your imagination and understanding, will be completely different. Okay, I may say Corolla labiate. I don't know what you will understand by being labiate. Okay, so that unless you confirm your specimen that with your herbarium and check all the herbariums, not only here. You have to, example, if you're checking here, if you're not satisfied, you have to again go to Calcutta herbarium. Probably most of the plants will be available there. Okay, if it is not there, then you find out who is the subject expert in that species and send it to him. Confirm him. Let that subject expert, he would have spent more than 30, 40 years in that one plant. He will know, he or she, sorry, will know. Okay, will know and immediately tell. Then you go for the new species. But nowadays, we jump. If I see some Corolla little bit color different and little bit elongation, I will forget everything about polyploidy, geographical isolation, ecological variation, everything I will forget. I see my Corolla is little different. Yes, for this new species, I won't ask anybody who has spent 40, 50 years in that expertise. I won't ask him. Yesterday I was born. Today I know taxonomy. Tomorrow I am going to put a new species. Here it is. Take it. You do whatever you want. Okay. Next. So basically, basically, before going for validity of a plant, I said a lot of things that you have to be able to understand. One of the things that you have to understand is different types of habitat, forests. Okay, and this we have already have in our walls here. Okay, next, so that you will understand this plant will be available in this habitat only. So it can be found in other habitats, or it is very restricted in other habitats. Okay. So what is, for example, in Sikkim we have tropical vegetation. Next, and these are the specific symbolic plants that you will find it in the tropical vegetation. Okay. Next is subtropical vegetation. Again, this kind of plants, those plants that will be available here. Next one, all these things. Okay, you will find it in subtropical vegetation. You won't find it in tropical vegetation, which is lower, but for 200 to 600 meters, 500, 600 meters is tropical vegetation. About 600 to 900 will be subtropical, 1000 subtropical. This thing. Next one. The temperate vegetation is about 1800 will come to temperate vegetation. In this temperate vegetation, next is this kind of plants will be there. You can see the marked gradual, there's complete differentiation of species of what is the species that is available in each type of vegetation. If you don't understand that slabs, then it is difficult to place your species particularly immediately. Okay, so you should know what is this thing. Next one. Subalpine, about 2500, 3000 meters, you have subalpine. Next, and you have this kind of vegetation. It's obvious it is completely different. Next one. Then alpine vegetation above 3000 meters, you have this. These are the species that you have. Next. And other species kind of, since we are so close to Sundarbans, is one of those, these are called mangroves. These mangroves have different adaptations. You all know, you have studied. They have vivipary, they have what you call pneumatophores, which are called knee roots. So this kind of, by seeing that itself, you know, you want mangrove plants, you cannot search it in Sikkim. Certainly not. Okay. You cannot certainly search a mangrove, get a mangrove Sikkim. So you should know what is the habitat, what is the type of vegetation, forest that is available. For that, you should know what are the forest types. And if you want to know forest types, I recommend a book called Forest Types of India by Champion and Seth. Okay. That has more than 60 or 70 forest types of India. You select your state and find out what are the forest types. Okay. So they will give what is the forest type and what are the representative elements here. Okay. Next. So these are all now some mangroves. Next. Next. And most important, I told you before itself, that to understand classification. So earlier you had to understand glossary. Now you have to understand that forest types. Now you understand classification. Okay. So in first you see that this is obviously taken from internet. It is this, you know what is phylum, what is the class, order, family, genus, species. What are these things and what are the groups of plants that come in these things. Okay. Next one. So for field work, we feel that Bentham and Hooper classification is more than sufficient in identifying a plant in the field and coming back also. Morphology based on morphological features. After that, you will have phytochemistry, molecular, metabolics, everything. Metabolic, whatever new technology that is coming. But on the field, and coming back to the taxonomic lab, to identify a plant immediately based on its morphological features, I find personally Bentham and Hoover classification is the best. So what is Bentham and Hoover classification? Nothing. It divides into flowering and non-flowering plants. And then in the flowering plants, it is said dicotyledons, which has 2 cotyledons, monocotyledons, which has 1 cotyledon. I think you would have studied this in some other channel, but recollect it, mug it up somehow. Okay, next. Today, I just show you the representative of Bentham and Hoover classification for flowering plants alone. Okay, I'm not talking about gymnosperms. So in polypetalum, it is very clear. Dicotyledons, you have 2 cotyledons. Polypetalum, it is what is polypetalum? Sepals, and the petals will be completely distinct. Sepals and petals will be completely distinct. You can easily differentiate. You can see here. Okay, these are all some of the. It has 25 orders, and in polypetalum, it is again put into 25 different suborders or this thing. The first one is Thalamiflorae, where sepals, petals, stamens are all attached to a receptacle. When you dissect the plant, you will see it is all attached to the receptacle, and you can this thing. Okay, this is Thalamiflorae. That's all. Very simple. And these are all the representative elements that I had captured when I was working in the field. Okay, next. Now, in same polypetalum, again disciflorae is coming. Disciflorae ovary will be superior. So to know ovary superior or inferior, how to find out which you have to go to the glossary. Okay. So then only you will know how the superior ovary can be identified. Okay. So once ovary superior is there, and the superior ovary will be immersed in the disc. There will be a disc in the petiole. In that, the ovary will be immersed like that. Okay, in that you have these kind of representative elements. Okay, you see, you can see the disc. Yeah, that's what I should do. Okay, this is the disc. Okay, next, and then calciflorae, you have stamens are fused to the calyx of flower. Stamens are fused to the calyx of flower. You can see all. If you open your Pisum sativum, you have very commonly open it. It will show that it is a monodelphous stamen. That stamens are fused, and the fused stamen filaments is attached to the calyx. Okay, that is why it is called calciflorae. These are all the representative elements of that. and gamma petal is inferior ovary, and also the petals are fused. Now, once the petals are fused, it becomes corolla. It is termed as given as a term corolla. So it has to the gamma petalae, the infrared, first this thing is infrared, which is having infrared. Infrared means ovary is inferior. Okay. Next, it has 3 orders. Okay, leave it. Okay. Then gamma petalae petals are heteromere. Again, the ovary is superior. Stamen in 1 or 2 whorls, carpels more than 2. So these are all rhododendrons are come under this ericales. Okay, or the ericales and this. Okay. Next. And again, in bicarpellate, the ovary is superior. There's a stamens is in 1 whorl and carpels also. Very clearly, no, that's not imagined. They have dissected almost 96,000 plants, and each plants they have noted the characters, and based on that each dissection and characterization, they made the classification. So if you see it in the field, it will be so beautiful. You have Benjamin Hoover in mind, and you dissect the plant or see in the field, it will be very nice, easily you will be able to tell. Okay. These are all the bicarpellate, which has 2 carpels. Bicarpellate means you have 2 carpels. And next, monoclamaria. Mono means single. That means there won't be any differentiation between petals and sepals. It will be all in same color or same form. It is called a period. Technically, it is called period. So these are all this thing. This monoclamaria has again into 1, 2, 3, 4, 5, 6, 7, 8, 9 orders. 9 orders. Okay. So each one order is having a typical character for that. Okay. Next. And next comes the third group in angiosperm. It is called monocotyledon. Again, only one cotyledon, and flower is trimerous. Okay, trimerous means everything will be in threes. Three periods, 3 stamens, 3 carpels, or 3 ovules like that. Okay, the first microsperm is ovary inferior. Seeds are very minute. That orchidaceae comes in that. That again epigyne. This epigyne will be your zingiberaceae and all. So on and so forth. Again, glumaceous will be the cypressaceae and poaceae. Okay. So each character, each order will have its own families. Okay. That families will have that order character of the orders. So you have to really understand that each read it. More than reading, if you are having field, you have to dissect the plant and understand the character. That will be more important than reading. Okay. Next. So that's one thing for taxonomy is again, it's not only the reading. You should do the field work. You should be in the field. You should then only you'll be able to understand the variations. Okay. Or else if you think if you're sitting in the lab for a whole day and then suddenly taking a plant, every plant will be new. It will be a new species, you'll jump and put the new species. I have a new species. Okay. Unless you have not thoroughly scoured the particular plant entire habitat variation, don't venture into new species. Okay. They call it today name noise, species noise in taxonomy. Okay, next one. So this field work along we have to go. Okay, these are the field work of SHRC. Next one. Next one. Next one. Everywhere you have to go. Then only you know the variations. Next one. Okay. When you go to the field, what then next? Means you will always carry a field notebook. In that field notebook, you have to have this data. This data is your passport data. Without this data, you can't. It is a very, very less chance of identification of a plant if you are a newcomer. Okay. If you are an expert, you will immediately tell. Where did you go? Will ask. You will tell this. Where is the altitude? What are then you will detect genus or something. But if you are a newcomer, don't venture for identification with this passport data. Okay, you should know, you should have all these things. Okay, next one. And this passport data will what we have in field notes, and we will transfer it to the herbarium sheet label. Well, this then your identification will be little much easier. Next one. Oh, this is obvious to tell. Nothing after collection, identification, or description. You have to cross check your specimen, or you have to verify the specimen always with the herbarium specimen, whether your specimen is matching with what is in the herbarium. Okay, herbarium is not entirely correct. Some specimen may be deciding rate, but majorly maximum no, what do you call probability lies in the sheet being the correct name. Okay, next one. And this is very interesting that see this is the last slide. Don't worry, I'll finish it here. But I wanted to say how innocuous thing can lead to species identification. Honestly, I am from south of India, and before I came here, I didn't never knew a family called. We never came across a family called Lardisa Balasi. Isn't it Norbu? Norbu was the first person. Sir, this is Lardisa house. So then I was just going through. Then I came to this. The top left corner is a moth, which is found very frequently in Nepal. Okay. Okay, this is called Agris salpuria. Okay, and this moth has a very specific feature. That means that the caterpillar of this moth will feed only in members of Lardizabalaceae. A near family near to Lardizabalaceae is Menispermaceae. If you feed that caterpillar, the Menispermaceae member, it will die. Any other plant you give, it dies. It survives only exclusively on Lardizabalaceae. Okay, fine. So this is one representative of Lardizabalaceae, which is found in Sikkim. Okay. In Thailand, there was on flora of Thailand when they were doing, there was no mention of Lardizabalaceae family at all. But one of the entomologists saw this Adresocria, and he sent to the person who was doing flora of Thailand, saying this is there, this exclusively feeds on Lardizabalaceae, but we don't have Lardizabalaceae. Then they again came to Thailand and embarked on the field tours, and then they found one Lardizabalaceae. This Tontonia parvatifolia. It's not here. It's Tontonia parvatifolia. It is up to till Arunachal Pradesh. We have it in India. So based on the identification of one moth, they could discover a new presence of a new family in Thailand. That is how nature is interconnected. Don't underestimate nature so easily. Okay, you have to, when you go to field and bring back all the passport data that you can take it from the field, then it will become very easier for you to identify the plant. Okay, next. Thanks. Anything you want to clarify?
No. Do you want this cable? Next is Govi's presentation.
After mine.
Achha, now it's all. Then you can switch it to your presentation. Here is the table, yes. Hmm? Ah, yes. You are presenting from here? Yeah, I'll present from here only. You want me to turn on my this camera also? Which one? No, 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 not needed. Yeah. Not needed. No mic, no camera, okay?
So that photograph has to be translated into a photo plate, just like this. So like this. This kind of photographs has to be translated into this kind of photo plates. Okay. So I'll be showing you people how to make this kind of photo plates. So for that we will be needing what we will do is dissection. We'll dissect all the plant parts. We do photography, and we process those photographs into in this kind of digital image editing software like Photoshop. Basically, we use Photoshop only. And how many of you know Photoshop? This kind of Photoshop, how to do? No one knows. So that if time will be there, I'll be showing a little bit how to do. So at first it started like this. There was no photography at that time, no? So it used to be only based upon the texts and all. And when there were no this kind of photographs, and even not the graphite were also not there, people used to draw this kind of things with natural dyes and all. This is from this Materia Medica from 50 to 70 AD by Thorides. He was a physician. He was a Greek physician. So he drew all these illustrations in his book. That is a 5-volume book. Mainly you reference as this medicinal book. So for medicinal plants, it has to be identified correctly, no? So he has uh, made all this type of illustrations in his book. And later on, it got like it became little bit modified with with time. It became like this. The pencil drawing, the line art, it became more like realistic. And after that, this is from 2015. This is more prominent. It looks quite realistic. This is a graphite illustration. Some Brazilian this artist has done, and this is what we get now. This is also called the image specimens. This is a composite. This is a composite image. What they do is they take a photograph in field and they even paint it, and then it will be edited digitally, and this type of plates will be produced. These are quite sophisticated. These kind of artworks are quite sophisticated. What we do in field that is quite easy and time saving also. Why are we doing this kind of things? No? See this specimen and this. These two are the same. This was collected in 1800 and I think 97. and this I collected in 2015. So 1897, there was no this kind of photographs and all. Photography was not there. So they used to draw it. It is a lot of time. It is a lot time taking. So the main problem with this kind of specimen is this is also an important part of taxonomy, but the problem for identification would be the color and the texture. The color, texture, and the indumentum it gets lost with time. The same plant. This is a field drawing. I drew this in the field. This is also a lot of time taking. Instead of doing this also we have to do. We have to do like this also, because without doing this, there is no point of doing this taxonometry. This also we'll have to do, but this is quite a bit of time taking. So instead of doing this, what we can do, we can take photographs in field itself. So this will be preserved for long term also. This can be preserved and this can be transmitted in any form, in any electronic form. This is also what I drew in 2016. This is the plate. This I draw from. I made this illustration from a specimen. The problem is the error, the human error. See this stamens. See how it looks and how it actually looks. This is not in proportion. So to check this kind of errors, what we can do is the best way. This is the best way to do. It is quite easy. I'll show you how to do it. See this here also the pistil looks different. No? This is what I have drawn, and this is what it is actually.
It's quite close.
That's all. I realized it afterwards, sir, after making this plate. So this is also another thing, the size of the plant. This is a giant taro. This is Leucophasia gigantea. So in specimen, you won't be able to see the size. No? Size scale is there, but you won't be able to see these textures and colors. The light, texture, and the colors you won't be able to see in this kind of specimens. So this kind of plate is quite helpful at that time. And this is the same plant. This is what we took in Manila last month, I think. This is Erythema graffiti. And this is the same plant drawn by one famous painter, this botanical illustrator Himalayan Region from Kalimpong. Sometimes we cannot take good photographs, and at that time, this type of botanical illustrations come in hand. This we won't be able to collect this type of information all the time. This type of tubers all the time. We cannot take in photographs. No? So at that time, this kind of botanical illustrations it helps us in taxonomical works, but we are not that much trained, and we do not have that time also, no, to make this kind of illustrations. So what we do is we do like this. This is a rhododendron illustration made by Walter Woodkey, and the same plant we collected this in last time in Manila, and this is the illustration what we have done. I think this might have taken like a month or so to make this slide. No? This one lithograph, it took us hardly like how much of time? 2 hours. 2 hours for photography. Maximum 2 hours of photography and like 20 minutes in Photoshop.
That is Usain Bolt saying 7.8 10 seconds I can run, but not everybody can run. So just take it with a pinch of salt.
What I mean to say, it saves a lot of time. What we won't be able to do like this, we can at least do like this. No? So this is the importance of what we will be doing. This will help to develop the habit of observing plants quite closely, and to see the plant parts much more detailed manner. And also it helps in writing description more accurately. The main goal is to write the description more accurately from fresh material. So because what we see in the specimens after collecting, that the specimens gets mottled and this discoloration happen. No? So in order to check that thing, what we can describe it more accurately from fresh materials. And this photo plates, what we'll be making in the end, this will be much more accessible and it will be a reference for future also. And it will help to substantiate your description, the plant description, what you'll be writing. It will help to substantiate that part. So and also it will help for easy identification plants in field for amateur botanists, for students like that, and or for other local people like that. And the main thing is it saves a lot of time. But this is not a shortcut. Definitely, it is not a shortcut. It has to be done very meticulously, and it takes time also. So the things you'll be needing is a digital camera, or either a digital camera, preferably, or a smartphone. Smartphone also it works, but you have to know how to use it at that time while taking. And a black cloth, velvet fabric is good, and the scale, the dissection kit. An ordinary dissection kit will do just one scalpel, a few forceps, and all those things. And a Fevicol or Fevistick, and this A4 size papers folded twice. Speaking of microscope, if you are bringing any small kind of plant materials into a lab, you can work out in this. And a desktop or a laptop like that, and a workstation like this. Application software, Photoshop. These are all what you will need. So you can write it down if you want.
No, you just share this PPT later on to them.
Okay.
Yeah, that will be better. We have to go a little fast because already we are delayed.
We have to do nothing. This for getting these photographs, we have to take all these kind of photographs, different parts of plants. All this photograph has to be taken. So how do we do that? Cloth, it has to be laid on a flat surface, and then you put your plant sample there. You take a photograph of this twig first, and then it has to go by step. You have to like go inside. You have to zoom into different smaller parts. From first the flowering twig, and then the inflorescence, and then the flower buds, bracts, these flowers, lateral view and front view. Also, you can take and this leaf, both the sides, capsules, seeds, anthers, the stamens, the filaments, the anthers, and all the petals. If it is gamopetalous, then you can dissect the corolla. If it is polypetalous, then you have to dissect all these different petal parts, all the segments. And the most important thing are the important characters and unique characters that are such as trap exudation and bark type. Like what Sir said, bark is also an important character. So flaking of bark, the lenticels present in the bark, those are also important characters. So those also has to be like photographed nicely.
At every place, this scale has to be there.
Yes.
In all the places, you have to put the scale. Otherwise, your whole things will be very messy. That will be misleading. Not get the exact the size of the plant or the parts.
So for getting a photograph like this, what you can do, you can make someone hold this velvet cloth in on the backdrop of your subject, and you can hold it, or you can just place that this velvet cloth behind it, and then you take a photograph just like that. It is quite of a trial and error. First you take one photograph and then see if it is coming good, then it is okay. If it is not, then you have to you have to adjust it in your camera, and then you have to make it accordingly. You have to adjust it accordingly. And in the end, all the dissected parts has to be like pasted in a paper, on a paper, with the help of glue. And then obviously the field number has to be there. What you will be collecting, that it will be attached to that specimen. And after taking all of these photographs, this will be processed into this kind of plates. This is for polypetalous because all these petals are like separated all these petal segments are separated, all these segments. And for gamopetalous flowers, what you can do is you can take a photograph of a whole corolla, and then you can even split it. If it is not splitting, then you can like totally split it up, not just cut it. And with those photos, what you can do is you can make a plate for description also. But this is a descriptive plate, and again this is also a descriptive. These are also descriptive plates, and this is a comparison plate. See, these are red, green, blue, and green are there. So this is for one species, this is for another species, and this is for another species. So you can compare all these kind of species with the help of those photographs. And also for labeling the floral parts, what you'll be showing in your work, this also can be done.
So for this, you have to know the Photoshop. So Photoshop, and then whatever you have taken in field, all need to be. You go back, not once. Go back. Go back. Go back. These also directly you can paste.
Yeah.
This because all the scales are sidewise. No? This also you directly can paste. Many people used to do that, but when you are doing something, that artisticity is also is required. Or maybe with this, some of the things are not good. Suppose you see these details in between the 2, 3, 5, like all these things are there. I can go to a plate. So here everything has been removed, and you yourself has put your own scale, making reference to that what you have taken originally. So it is completely artificial you have made, but all the characters are retained there. So that's why the scale is very important. Next.
Like this.
Yeah. And whatever the detachment was there, that you keep as a ready reference. It is a double folded small paper, and that will be remain there. The specimen what you will be collecting and making the herbarium specimen, possible this you paste there also. That in future somebody will be doing. So this will be always attached there. That is the way the Hawker and British people used to do like that.
And lastly, what not to do, what to omit while taking these photographs. You have to check whether this backdrop what you are using is not multicolor. It should not be multicolor. It should be unique. I'll show you why it is like that. It should always be unique. Either it has to be completely white, black, green, or blue. It should not be multicolored. And the backdrop should not be as the same of the subject. If the subject is green, you should not be using the backdrop as a green cloth. Green cloth you should not be using. And also you have to check whether there are, like, in a background there should not be any superfluous things like vehicles, boots, houses, or plastics, malls lying like that. No wires, those should not be there. And also unessential matters that gets stick or attached to the specimen what you will be dissecting, there should not be such things like debris, grease, dust, all those things will be there. It should not be there. And also you should not be taking this broken or incomplete or infected wilted plant parts. It should be fresh. It looks good. It should otherwise it will be like it will not be good. It doesn't look good. And it is misleading
the
also.
actual character.
Yeah, it will be misleading.
It will be little bit vague and misleading.
And this subject, what you will be dissecting, it should not lie too close to the scale. It should always be like little bit apart from the scale. Last time, actually got it. This some of our friends have done like that. They have placed this subject on top of the scale. It should not be like that. And overexposing and underexposing of subject. It should be well lit. There should not be like any burning of images. It should not be overexposed or underexposed also. It should be adequately light. And also to check, you'll have to check the shadow. How you will be using your flash and your camera. That will depend. It will depend upon that. The shadow should not be there because if you cast the shadow over an important character, that will not look good. And focus should be there, always should be there on the subject. What you will be taking a photo, what photograph you will be taking, the focus should be there. You cannot use a blur photograph. And plant parts should always it should be accompanied with a reference scale. See, this is a mistake I made while I was just learning this thing. I just took all these photographs without any scale, and after arranging all these things, I came to know I do not have any scale, and this is useless. That much is there. And so should I show how to dissect all these things in Photoshop also?
Okay. So
I'll take-
it's okay. Already we are late. What is the time now?
Of course.
Yeah. Okay. So if anything is there, you can ask.
Yeah.
Anybody
Otherwise,
have
what we
any
can-
question, or otherwise you people keep contact with us while doing all this thing. Yeah. Some people are actually there already from these participants. They are already working with us, and if for something also you can make some collaboration with us. Okay. So any further assistance is required or further something is there, we always will be there. We are open to everyone. We wanted to because you see what is our objectives, and that is we are trying to do that. What it is said that taxonomy is a little bit like that only. Why this thing is required? The most fundamental question is that these type of things do not have any human error. A illustration which is line drawn, it will I will see different way, you will see different way. So maybe you cannot communicate it directly while going to the field. The plants, maybe you may think that this is something like that. But when you will see these things, if you will see it in the wild, exactly what you are seeing in the photo plate, exactly you will see in the wild also it is in plant. Okay, then that will be very easy. That's why this is some of the few years before the team which is going to be presented here in the next half. Abhishek Bhattacharya, one among them, who has actually in BSI they have brought it, and we are now all in following. Even seeing us, there are some of the Western people in Kew and Edinburgh also they have appreciated. They have followed us. It is not like that earlier only Western people we used to follow them. Now a lot of Indian things has been followed by the other world. So like that. Even why Tamang sir was here today, you also would be very happy that a lot of things that what Tamang sir has brought out that has been appreciated, and people are trying to do those things. Even it is commercially or economic utilization also can be there. So if anybody wanted to ask further something, you can ask, no problem. So then it is done now.
Yes.
Yeah, then I will go for my thing. Okay. So sir.
हम डाल देंगे।
वहीं से रन कर दीजिए।
Identification करने का।
Identify.
Wait. This camera, I have to use that one.
हाँ sir.
I have to use that from there. I have to speak, or yeah, from I can do.
From here only sir you can do it. वो उसको presentation mode में डाल दीजिए मतलब. आ रहा है.
Yeah. Okay. Good afternoon, everybody. I'll not take much time here. Just I wanted to show something like that. See, this is what you have already read in your theory classes, your MSc or something like that. No, I just wanted to show this is what our intention to make this program, and this is in series we will go, because some of the people, I think 17 students already have joined our first program. This is the second program, and like that we wanted to go for further. That for giving this idea about this taxonomy and something like that. First was ecology, something like that, quantification. So history can go to the next slide, please. Yeah. See, you know how this classification taxonomy has started. The Theophister was the first person, a big philosopher. I will not speak anything more about this. Let's go to other thing. This is very important. This is why it is very important. We always think that everything happened in the Western soil, but it is something not like that. It is, I already told in the morning, and Tamang sir also told how Joseph Dalton Hooker's visit to Sikkim and Darjeeling was important. It is not for only our place. It is for the giving a light to the mankind, to the whole humanity. So here you see, this is a ruler. He was a born reedy who came and settled there in Malabar, which is Malabar coast. It is in Kerala and those places. and he decided to write a book, Hortus Malaveritas. Whatever the plant used, that, and this was such an important thing. It was, you have seen, it is 1678. So when we talk about taxonomy, our last line that is actually 1753. Why 1753? Because that is Linnaeus and those things have happened. It is before the 1678, which much before Linnaeus. These activities happened in the Malabar coast. Although he was a British fellow, or he was a Dutch fellow, but the people who was involved for preparing this book all were Indian. He brought all this medicinal man, healers, and those things like that. Our ethnobotany and pharmacology and pharmacognosy students now go and ask how you treat all those things. So this has been done, and those were the basic material for bringing this whole plant taxonomy to a proper scientific subject. And this helped, and the illustration which was there in that book, those become the fundamental something for naming some plants. Many plants, not only some, many plants. Next, please. Next. Next. So this is that first page of the book. Next. See, then Kerala's Linnaeus has come. So what he started the binomial nomenclature, everybody you know, and that's why that you see 1753. That names which has been named before 1753, those do not qualify the modern taxonomic principles. That's why those are actually after these 1753 only, which we usually consider as they are the most authentic name of scientifically following the binomial nomenclature. Next, please. So this is the space plan. Next, please. So afterwards also Augustine Pyramid, this Sikandol, all those people have come. Next, please. So you know there's natural system of classification, George Bentham and this J.D. Hooker. So he is Hooker, which morning you have talked. Then next, please. So this is the natural system of classification main thing, and Dr. Franklin also might have spoke today. This is actually some based on some similarities of the characters like that we used to group. So this is same character, so this will be grouped here like that, and this is nothing with that what is the molecular and DNA and all those things. So next, please. So this is here from that what is called phylogenetic system of classification. You know all phylogenetic system of classification, which can tell the phylogeny how it is related. It's like that. It is slowly that molecular and DNA, all those things has come. Next, please. So this is conquest. Next, please. So very importantly, after 1998, this is the system has come. What is this? APG, you all know, I think. APG, Angiosperm Phylogeny Group. So this is not the one or two people. It is a group of botanists and scientists came together to classify the whole world's plant on the basis of what? This is the DNA sequencing and those things. And you all know it initially came as 1, then 2, 3, 4. Because why? It is a lot of revision is required. Whatever they are putting, those things still need revision. Next, please. So you see why identification has to be very correct. I told in the morning that if somebody is doing, and sir also told that if suppose somebody is doing with some plants, if that will be not correct with that, with the finding of some new novel compound, somebody will attend. Or some you have got some medicine that which can cure diabetes, which can cure cancer. So that time these things will be there. And if your plant is not correct, then that will how much problem that will create complications. You know, you are working with a plant, and if the name is not correct, if suppose there is some toxicity, the plants which you have given the name, but that is not the actual name. Your plant, if you have wrongly put, it has the DNA damage activity. Then people will have, if that medicine will be used, then people may have cancer. Rather, that will cure the disease. So this is that how serious the problem is. Here you see, this is a plant collected by one of our scientist, Joseph, J. Joseph, 1968 from Arunachal Pradesh, and he has simply named it as Impatiens amplaxicolis. Okay, seeing that here is the amplexicol leaves. This is the plant. Here also amplexicol leaves. But When later on one Chinese scientist very carefully examined, and he found that the plants which is found in Eastern Himalaya Amplexicolis, this is not Amplexicolis. That's why he named it as Impatiens summensis, a plant from new species recorded from China in 1978. But Indian scientists has collected that plant in 1968. How many years before? 10 years before. Because the Indian scientists did not study it properly and correctly not done. That's why this fellow did not got anything. He was getting a gold coin, but he thought this is nothing but aluminum. He thrown it. That's why the Chinese scientists got that gold coin. Next, please. See, same. Here also, this is a plant, Rola Sosagri Rao. He collected it from again Arunachal Pradesh, this plant. And he has written it in Impatiens bicornuta. Okay. So here also the same thing happened because he did not study it properly. And then when Saurabh Jyoti Bora from Guwahati University, who was earlier in Botanical Survey of India, and myself went to Dalai Valley and we got this plant. When we have very carefully examined, we found that this is not bicornuta. This is another species not yet described yet. So that's why it became Impatiens dalaiensis, because this is we got it from Dalai Valley. Next, please. So same thing, D.V. Dev in 1964. He got this plant and he identified it as Impatiens lutea. And Saurabh and myself, we got it from again Lohit Valley, and this is Impatiens lohitensis 1912 disease, or no, 2012 we have described it, and it was in 1964. This plant was discovered actually first in 1964. Next, please. So same seeds you see, Joseph, 1964 he collected from Odela and identified as Impatiens longipes. And this linen came from Sweden. In Arunachal Pradesh in 2017, Fungus Bharali, they have made it a new species because the same plant. Actually, these plants, when they have published, then I got it. Then later on, I have checked and I have found that this was far many years before these plants was collected, and this was all wrongly identified. Next, please. The same thing you see. Why this is happening? This is happening because of this. Even from a layman eye, if you see, both the plants look alike. Okay, but that is Impatiens lohitensis and this is Impatiens stinentha. Next, please. So here also see the same thing. Many people like that. This is Impatiens divangensis, that is stinentha. This is stinentha. This is divangensis. And see now, when you will be seeing very carefully, you see here how this formation is taking place, how these things are there, and how these things are. So these are the things that you need to be done very carefully. Like same here also. And again, here you see many times the plants looking very similar. But if you see the fruit, here you see this is very linear, it is clever, it is going up, and it is something like that. Different. Next, please. So same thing here also, one thing happened. This is Impatiens levigata. This was also in many years before, I think some. So when we examined it, then we found that this is not levigata. It is similar, partially similar, but it is pseudo levigata. Next, please. So what, now you see this is pseudo levigata and this is levigata. Okay. So it is seeing similar, but you see how the break, this lateral sepals are here and how the break is here. So here you see the breaks are something like this here. Lateral sepals are like this, and here you see these two small lateral sepals are like this. Here the lateral sepals are like this. Okay, and not only that, this here you see the span. So this way, if we don't see all these things very thoroughly, then the complications and those things we will be not getting that gold coin, but rather will be a waste. Next, please. So characterization. So this what Norbu has already shown. This is the thing I told you, the scale, dissect, and all those things will be there. Then this will be your reference. Next, please. So like the same only. Next, please. So this is that after formation of the plate. Next, please. So after dissection of all these things, this is the thing that what he has told to keep like that, keeping in a scale also, and this for your ready reference. If this plate is there, if you are not keeping the scale, it has to be there. It is always like a ghost like that. If people, if people, some know. before going to the species, you can go, maybe the genus is again subdivided into small groups. That may be subgenus or section. So here this Musa is globally divided into 4 sections. That is called Musa, that is called Kelly Musa, that is called Rhodoclemis, that is called Australia Musa, and one another classification also was there, that is called Ingenti Musa, but that has been reduced. And now, on the basis of molecular phylogeny, now the scientists are telling there is only 2 sections. One is Musa, another one is Kelly Musa. Even Australia Musa is also been reduced to Kelly Musa. So then, when we talk about the Rhodoclemis, we have seen that what is the characteristic. Suppose let's leave it this molecular thing, but Rhodoclemis and Musa, what are the characters between the Rhodoclemis and Musa? So then it is defined that when it is Musa, the inflorescence will be always patterned, and in Rhodoclemis, the inflorescence will be always erect. So in my plant, it is both the characters we have seen. It is inflorescence erect also, and sometimes pendant also. That's why when this on the basis of molecular, it has been reduced, because this, I think, both the character, and this is sometimes when one character is dominant, then it becomes independent. Sometimes it is something like that. So this is like the Rhodoclemis. We got some species. There are some 7-8 species, and we have seen the types also. We have seen the specimens also. We have seen the live plants also. Then we found that one which we have seen that is not matching with anything. That's why we have impressions marked with. Now next please. How I reach to the family? Now I will show you that. See, when we came up to the Cytaminus or G-Virus, then we found that G-Virus is the Moses, okay, and Heliconiaceae is also there now. We will not go; those things are Latin America. It is Brazil and those places it is found. So whether the plant that is matching with this, the vegetative plant coming there, very different. It is not coming to this, so this cannot be gingiva. Okay, next please. So this Kanasi from Kanna, it is also very different. It is not coming fruit. Kanasi, this is not. Kanasi. This is also more close to Kanasi and gingiva, but not the plant what I have got. Next please. So this, whether it is having similarity, yeah. Out of all the families, it is having highest similarity with this plant. So it means that plant what I got, that is of which family? So this is clear everybody now? It is Musaceae. Now next please. So how to reach the genus? Already I told 3 genus in Musaceae. Next. This is only found in China. It is nowhere in other part of the world. It is, and I have seen also. You people also not seen. I am also not seen. From some. So this is how different. This is one very big trunk, like the stem is coming out, and then some this, and it's not a very big plant, but something 2 meter up to like that. This is Musella. No, this is not a plant. Similar. Next, please. This is you people see. You have seen? Mamta, you have seen. Okay. So this is Mamta knows that this is NCT. Why is NCT? Because it is similar to that plant, but it has the persistent brake. It is like banana, but it is always the brakes remain with endurance. Okay. So banana like small banana. That's why it is a separate genus. NCT. Next, please. So this is, yeah, it is not having similarity with. So what genus it is? Now see, under Musa again, the 4 sections has been in India, 2 sections. Of India, this Australia Musa and Kelly Musa was there. Kelly Musa, those are in Borneo, Indonesia, and those places is very. And this Australian Musa also same like the islands of Australia, Australia mainland, like and in Rhodoclemus. Rhodoclemus and Musa, Rhodoclemus and what volume will be as Musa. Next. So this is no that you go back. Inflorescence will be rhodoclemus. The so whether this upward or it is so this is all event. This is event. This is rhodoclemus. Next, go back. Go back. This is Musa. Okay. Let's see, just follow the whole class. So whether this is upward or this is any rhodoclemus. This is Musa. Okay. So let's see, just follow the whole classification. Then that is, so this is Kelly Musa. My plan is not like this. Okay. Next please. So this is Australian Musa. This is in Borneo, all those places it is found. Next please. Papua New Guinea. Then next please. So see, these all are rhodoclemus. This is we have a plant, we have a collection here also. You have seen this plant? All have seen. You know what is this? No, this is Musa velutina. Okay. So whether it is matching with that plant? Is it matching with that plant? Yeah. Somewhat matching, but not 100%. Next. This is Eventiaca. This is somewhat matching. Okay. It is somewhat matching, but still you have some doubt because here fruits are different than this male bud. It is called male bud. The where from this male flower and these things are coming. This is not exactly like that. This is with purple, and that is pink. So you have doubt. So next, please. So whether it is matching? No, it is totally different. This is Chuni. It is in China, in Arunachal, and in China and Myanmar it is found. Next, please. So it also has some similarity. Next, please. This also has some similarity. Next, please. This is no. This is not from here. It is from Vietnam. Next, please. Go back again. Yeah, go back. See, just wait. Now, which are the most closest species to that plant? What we have seen. You go to that plant. Go the first we have seen. Go back. Go back. Go back. Yeah, now you keep it here. You see the plant very carefully now. You have seen, no? So you have seen here. Then you see this is the bracts are basically these are all pink. Okay, and see the shape of the fruits. These fruits these are quite bigger. And number in this is one. This is whole thing is called one bunch, and these are called one hands. And one hands. There is sometimes in rhodoclemus only one row. In many in others 2 rows of fingers. These are fingers. One fruit is called finger. So one row of fruit. In some 2 row of fruits. Here 1 row of fruit, not 2. Okay, but the number of fruits are also here in one hand is more. Now go again down. A little bit, yeah. See now whether it is matching. It is somewhat similar, but not. Next. If you read all the characters, then it is not exactly that. Then again go back. Go back. See, here you see, this plant is the most closest allied to that plant. So that's why initially I told if all the identification has to be separated from the most closest allied from that species. So this is not matching. Why it is not matching? But because these fruits are quite different. then those fruits are quite different. And it is purely purple and that is pink. So this plant can be separated, that plant can be separated from here. So in that cup plate leaf, then what is the contrasting character? These two plant will come at the final. Okay. So those all the characters. Here what will become? The male bud purple, male bud pink. Okay. Purple and pink already is a contrasting character. Then comma. Then size of the fruit. Suppose that is up to 10 centimeter long. It is up to 5 centimeter long. Organism it is 5. Highest is 7. That is not 10. Okay. Like that. And some other characters also will be coming. Like if it is suppose the ovules, here it is 2, there are ovules 4, and locally it is different. So when there is some anomaly all those things, then you can go for a DNA sequence. Then it will clearly will tell whether it is different or not. That's why many a time is a lot of species are got reduced also on the basis of that. So next, finally. So now you see, this is the ultimate judgment. What happened? This is Cytamini you can got, Musesi, it is not Jealousy cross, not Kanasi cross, Mayatesi no. This is Musa, this is not Musela cross, NCT cross. Yes, this is Rhodoclamis. This is Musa no, this is not Kelly Musa, this is not Astili Musa. This is that purple plant was Musa ornata? No. Then Musa erotica? No. Musa velutina? No. So this is Musa, it's a different species. So this is all about how we have to read the keys. And this is how we can go for identification. It is not like that simply you cannot go and see that what is this. That blindliness will create the disaster. So we have to have the systematic way of approach, how we can reach up to that species. So this is what the plant identification is. Okay, thank you very much. So what is the time now? Now you all people, I think already very exhausted. So the lunch is ready. You can go to our type 5 quarter there. So their lunch is ready. After that, we again will assemble, and next session will be very rigorous, and that will be fully from there, from Calcutta. Okay. Yeah.
So my presentation is visible. Okay, thanks. And I'm audible as well. Okay. So good afternoon to all, dear participants, faculty members, my colleagues and seniors. So in previous sessions, you have been experienced the three nice talk on the plant identification, some of the classical literature, and some historical account of the nomenclature and etc. So today, in this talk also, again, you may have the experience the same thing in repetition, but in the different way. Well, if we talk about the identification is nothing. It's just a, you may say that matching with a non-species to a unknown specimens. So it may be to any rank or position in extent to the classification. It involves the finding a name for an unknown specimen. If the identification result is negative, that means your specimen might be new to science or a geographical area. मैं इसमें ये कहना चाह रहा हूं कि कोई भी स्पेसिमेंस जब आप कभी आइडेंटिफाई करते हो, अगर नॉन स्पेसिमेन के साथ जब हम उसको कंपेयर करते हैं, if the result is negative, that means that specimen can be new to the geographical area. Say for example, if this plant is not matching with the collection from the Sikkim, then this will be the new addition for the Sikkim. Don't say that this is the new species for the Sikkim. Or oftenly I have seen in most of the students are committing such silly mistakes and they are saying that this is a new species. No, this cannot be a new species because this is already known from the some different place. So you may say that this is a new to the geographical area. And if this species is not matching with the existing entire world specimen, in that case, you may say that this species is the new to the science. So this is the basic differences between the new to the science. Say for example, if you see the some of the article on the Globa racemosa, some of the students has committed a mistake. In India, what they did, they wrongly said that Globa racemosa is a new species from the Assam, and somewhere they have published. So our purpose, don't do this such kind of mistake. It's very interesting that why we are going for the identification, and it may be the very easy task. No, you know that as a specialist who can identify living organism by heart, like Linnaeus, he can remember the thousands of names very quickly. Likewise, especially in the tropical country where the biodiversity is huge and high, and taxonomists have to develop some of the kind of tool to reliably identify their materials. You know, at present, there are in around 36,000 of the species described so far. And can you remember all the names? It is very difficult to remembering all these names. So we go for the some of the technique where we can apply such kind of things and we can at least know the family or the genus level, or in some of the cases, the species level, we can identify. Now question comes whether to know this identification or the classification, education matters. Just see the one very renowned face, this John Hutchinson's. If you will come to know about his education, he was hardly high school pass out, and we all remembering him for his system of classification. Just to imagine, a high school pass out person can give a system of classification, and we have the lots of, and those time the literature was very limited. If this is possible, we can also make. We know that literatures how to extract from the website nowadays. Many things are available, online resources. It is not like that you have to depend upon some of the library, some of the university. Those days specimens was not visible, but at present it is visible through the virtual herbaria. So if it is possible, in that way, plant taxonomy is very easy science as of now. There are three basic methods to identify the plants. By using the flora, some of the literature like revisionary articles, and monographs. Don't confuse with the revision and monograph, both are the different thing. Some other day we can give the talk on that. Then another method is the herbarium or physical specimens collections. What is happening when a student from the university or a researcher is coming from the university or the institutions, they're bringing some of the physical specimens. And herbarium like Central National Herbarium, which is the largest herbarium in India, having the more than 2 million collections. What they are doing, they are matching the specimens with the existing collections. So what is the actual problem with the matching of the herbarium specimens? Many of the characters, which is very important, like in Orchidaceae, like in Rosaceae. So there are so many families where micro characters is having the significant role in identification of the correct species. So in that case, this herbarium matching is not a good practice, what I know. So if we will ask about the technical identification, means what is the correct identification, I must suggest that you should apply use the flora or revision or the monograph. And if both are not working, in that case, you have to ask the expert. As of now, many experts you may have take the help through the online also, but in past it was not very easy. In past they were having to depend upon some correspondence through the writing the letters and all. But these days through email you can easily access the person. Now question comes, how to use the flora and revision, how it can be helpful. So I am showing one of the key where you will find that this is very useful. This is Sapindaceae is the one family. This Anacardiaceae is the one family where you will see that how we have prepared the key. Leaves simple. If you are knowing the definition of what is the leaf simple, easily you can segregate the plant simple in a one group. How it is possible? Say for example, you are visiting one of the forest area. You have collected lots of Anacardiaceae sample. Means I am talking about how to segregate the genus, not to the family. For the likewise, the family having some different concept. So likewise, genus having some different concept. After the identifying up to the genus level, you have to go for the species level. So hierarchy is here. Firstly, you have to identify up to the family level. Then you have to go for the genus level. Then you have to go to the species level. If it is species also there, then some of the infra-specific level you have to check. So the key is very essential and very important part of the identification of a plant. Here I am just giving one example where you will see that in case of Anacardiaceae, वो जो मैंगो वाली फैमिली है ना, वो अनाकार्डियेसी में आप देखेंगे हियर क्या करना है कि दो कैटेगरी आप बना लो. 10 कलेक्शंस हमारे पास हैं. One group leading to the leaves simple. Whatever the leaf simples, they will be grouping in the one and group A. And another group where you will find the leaves having the compound, trifoliolate or the pinnate. Then this two leads to the here. So in leaf simples, there are the two options, one having the leaf opposite and leaf alternate. If it is the leaf opposite, directly you go to the Dobinia. Without any hesitation, you just go for the Dobinia. Likewise, if alternate, then again there is some cases are here. Like here, leaves crispate and with the distinct marginal veins, then it will be the Dreamycarpus, and remaining it will be go into the Mangifera and the Semicarpus. Likewise, for the leaf compound, again, you have to see that here, leaves trifoliolate and leaves pinnate. This is the cases of the compound leaf, where if it is the trifoliolate, directly go to the rus, and if it is a pinnate, then again there are the different genera like Pegia, Aspondius, Linea, and rus. So these are the some of the cases. Here now question comes, why it is written here the rus paniculata and rus is the 2 times? Why? The reason behind rus this leaf trifoliolate is the only one species here in the northeast India where you will find it is the trifoliolate. Otherwise, it is the pinnate only. So that is why it is in 2 groups, rus in the pinnate as well as in the trifoliolate. So going for the using the key, prior to using the key, you should have the knowledge on how to describe a plant, means what is the morphological description of a specimen. So you have to start with the habit, then you have to go for their stem and the branches. Then in stem branches, not like that only you will write that stem is the 20 centimeter long, not like that. you have to give the all the micro morphological character as well. Say for example, if a stem having the glandular hairs, you have to write a stem glandular pubescent. If a stem is the terate, you have to write the stem terate. If it is a cylindrical, you have to write a stem cylindrical. So likewise, you have to add on the characters. Similarly, for the branches, you have to apply the same. Then after the stem and branches, the terms comes for the leaves. Here you will find the leaves may be the simple or the compound. Again, if you are having the very limited description, in that case, you can write the leaves simple or compound, whatever. Some of the cases, like in the case of Asar, you will find that both cases are there. So you have to mention clearly leaves simple and compound both. Then you have to start with the base, margins, apex, textures, vein, mid vein, and the lateral vein, petioles, stipules. So this will be the sequences for the morphological description of a leaves. After that, we will come to the inflorescences, where you have to write about what is the type of inflorescence. Inflorescence means a group of flowers arranged on a axis. That's called the inflorescence. तो inflorescence में हम लोग उसका टाइप पहले लिखेंगे, whether it is a raceme type or ये cyme type है. Then we will write about the peduncles. You know that peduncle किसको बोलते हैं. Then we will go for the bracts and bracteoles. The basic differences between the bracts and bracteoles, in fact, both are the same. But for the distinction purpose, first time whatever the structure is appearing on the flower, below the flower, we will write it as a bract, and second, after that, whatever the characters will appear there, that will be called as the bracteoles. After that, we will go for the flowers. So flower may be the unisexual or the bisexual. And if it is the unisexual, you have to write about the flower unisexual. If it is a bisexual, it is a bisexual. Some of the cases, polygamodioecious cases are there, like in the Mangifera or the Anacardiaceae family. Then in the flower, you have to write the characters about the calyx, corolla, stamens, pistil, style, stigma, etc. Then you have to go for the fruits and seeds. Now question comes, why I am giving this much emphasis to writing a correct description? Answer is, if you will check the key, you will find out some of the, if the more than 30 species or the 20 species, then you have to depend upon the smaller characters to differentiate between the species level. So if you don't have a good description, you cannot assign a correct identity of a specimen. Basically, we are using this plant glossary of the Wentzsche, Henk Wentzsche. It's an excellent book. It is not available online, but you can access some of the good library where it is available. These all already have been shown, so I am not going into the details of that. Yes, there are some of the characters which is differences are very limited, but we are not able to define what it is in fact. So say for example, glandular means covered with the gland with a zone of the secretion producing tissues. A species, a sample leaf or the stem, it may be the glandular pubescent or gland dotted. The differences is here. If there will be the stalk, you will say that glandular pubescent, and or the glandular glandular pubescent. And if there is a sessile gland on the leaf surface or the stem surfaces, you will say that glandular punctate. Then if it is the gland bearing, you will say that glanduliferous. Then again, there is a similar terminology, puberulent, puberulous. The basic differences between both of these: minutely pubescent, the hairs hardly visible to the naked eye. If it is not visible through the naked eye, we will say that puberulent. And if it is visible and if from the touching to the surface, you can easily feel that this is the pubescent in nature, you will say that puberulous. So this is the basic differences between the puberulent and the puberulous. Likewise, pubescence and the pubescent. So if it is the hairiness nature, you will say that pubescence. And if it is a densely fine short hairs, downy, then you may say that it's a pubescent. But usually, if you are writing the pubescent, it is not a wrong thing. It is a good only. and but you are not defining whether it is the glandular pubescent or this is the scabrous or what kind of nature of the hairs. It is not defined here. So pubescent is a regular term. Then these positions already known to you. Most of us having the lots of doubt on the color characters. Even if it is called the mauve, we will say that purplish. So there are many specialized terminology for the color also, but we are not applying them very accurately. So better to know the exact term for the each and every color, and you should apply that correct way. This another foliate and foliolate. If I will ask you a plant having 3 leaflets, we have to say that the plant is trifoliolate. Foliolate means compound nature. If I will say a plant having 3 leaves, then we will say that the plant is trifoliate. So basic differences between the foliate and foliolate is the nature of whether it is a simple, then it is a foliate, and if it is a compound, then we will say that foliolate. Here you may say the example. If the leaf is the simple, then it will be called as the petiole or the petiolate. Leaves is the petiole, leaf having the petiole, and it is the petiolate. So if it is the compound, then you will say that petiolate, and the leaflet will be called as the petioles. This is not very easy terminology. Basically, when we are identifying the papilionoid plants in general, in legumes in general, there is the very big characters. In fact, pedate and pinnate leaflets. I don't think very oftenly it is coming, but in case of the leguminaceae, you will come across with this terminology. So the basic differences between these two terminology: if one leaflet petioles are the bigger, longer in size in comparison to the lateral 2 leaflets, then it will be called as the pedately compound leaves. And if all 3 are the equal in the length, then it will be called as the pinnately compound leaflets. So this is the basic differences between the pedately and the pinnately compound leaves. In fact, both are the compound nature, but in one case you will find that the petiole size is the bigger than the rest of the 2 leaflets. In case Vitaceae, you will find that in tetrastigma having all leaflets mostly are the trifoliolate or the 3 to 7 leaflet, where pedate term is the quite common. But in case of the legumes, rarely you will find this terminology, but it is present there. So in key, you can apply these 2 terminology to make the 2 bigger group. So these are the regional floras like Flora Bihar, this Botany of Bihar and Odisha, Bengal plant. And for the family, I will suggest that you should use these flowering plant families of the world. Then if you have some specimens, you can consult the herbarium. Nowadays, physical specimens and digital collections both are available. Like Q herbarium, you can access this. Very easily it is available, and you can access this. Nowadays they are not providing us downloaded images, but online you can see the specimens from the queue. So this is the one of the example. You will see that this Roelia specimens are there available from the Kew Garden only. Then if both are not working, then you can ask to the expert. This is the basic things what we are applying to identify a plant. But when we are looking to the means bigger area, then the students facing lots of problem. So for them, I have prepared some of the slides which will be encountered during the practical as well, and I will explain some of the characters which is very important. Like if you are looking into a bigger area like forest of the Neora Valley National Park, then Singhilala National Park, or the Buxa Tiger Reserve, or any biosphere reserve, something like that. In that way, you have to use the Bentham-Hooker system of classification, because as of now also I'm saying that even though APG4 having the 416 family, but the 202 family of the Bentham-Hooker system is the more convenient to identify a plant. So we cannot avoid this system of classification. Doesn't matter, it will be come whether this APG5, APG6, APG10, numerous number, but this system will be remain as such for the identification purpose. So what we are doing here, first job is the grouping of the plants. This is very simple. What I will do here, angiosperm and gymnosperm, it is known to you. Then in the angiosperm, the plant can be either to the dicotyledon and the monocotyledon, already known to you. So if I will talk about the gymnosperm, here you can see this one, this Pinus species. Here this sperm cone is, this is naked, and here you can see this seed is the covered. So this is the basic differences between the angiosperm and the gymnosperm. Then in the angiosperm, there are the three major group. One is the this Bentham-Hooker has the class having three classes: dicotyledons, monocotyledon, and in between they have placed the gymnospermy. So in dicotyledon, there are the three orders like polypetally, gamopetally, and the monoclemidae. So what is the differences between these two? These three. Here, if petals are free, here you can count 1, 2, 3, 4, 5. So this will be called as the polypetally. And gamopetally, here petals will be the fused here. You can see this corolla tube is the longer than this lobe. So this will be comes under the gamopetally. And if there is no distinction between the sepal and petal, and perianth will be in one or more than one holes, in that way it will be called as the monoclemidae. So in Bentham-Hooker system of classification, if we will start from the Ranunculaceae, this is the family number 1, up to the Caprifoliaceae, this family will be comes under the polypetally. From Caprifoliaceae to the Lamiaceae, all plants will be comes under the gamopetally. Then this Nyctaginaceae to the Salicaceae, this family comes under the monoclemidae. But very funny things happened, like Flecausea. In past, I have seen that some of the specimen has been shifted to the Salicaceae. But it is unfortunate that APG is doing something which is not like that acceptable. In polypetally, so these techniques you can apply. If you have the lots of polypetally, you just go for the series. What are the series here? Thalamiflory, where you will find that sepal, petal, and stamens are attached to the receptacle. In disciplory, that means there is the disc present, and calciflory, that means flowers are the perigynous or epigynous. In thalamiflory, अगर हम thalamiflory की बात करें तो उसमें पहला फैमिली हमारा आता है Ranunculaceae. तो यूजुअली Ranunculaceae कैन नॉट बी ए ट्री. तो जैसे ही आपको अगर कोई ट्री स्पीसीज मिलता है, आप पहले ही समझ लो कि Ranunculaceae नहीं हो सकता है. Simply either it will be the annual or the perennial herbs हो सकता है, some of the climbing shrubs हो सकता है, it may be the woody as well, or it may be the twinner. तो अगर हमारे पास ट्री स्पीसीज है तो एक फैमिली हमारा बाहर हो गया, means out of 202 families, not 202, 166 family, because I am talking about the dicotyledon only. Out of 166 families, the rest of families has been eliminated from here because it is not a tree. Ranunculaceae can be the only herbaceous or the woody climbers or the climbers only. Then the number of stamens will be the many. So from very primitive characters, you may say that if the stamens are more, that means it is the primitive. So in that case, here in Ranunculaceae, you will find lots of stamens are there. See the example. This is the one of the example where you will find it is not visible, but here you can see the lots of stamens are visible. Some of the time, petals are absent in case of the Ranunculaceae. Then, if it is the tree species and you have the stamens are the numerous, the plant may be the Delineaceae. So here, how to identify? You will see here the sepals are the very fleshy, and if you will test, that test will be the little sour in taste, and number of stamens you can see that. So thalamiflory characters are available here, but here no disc, but number of stamens are the numerous. So this is the identifying characteristics of the Delinia. Another character, even though in vegetative condition also you can find out in the Delinia indica usually, where you will find the leaves having the very prominent nerves. Magnoliaceae. In previous, previously it was there was the two genera usually, this Magnolia and the Michaelia, but as of now only Magnolia is present. Here you will see that usually these are the tree and shrub elements, and leaves usually the coriaceous. Flowers are either axillary or the terminal, solitary, bisexual, enclosed by the deciduous spathe like the bracts. The fruits here you will see that carpels dehiscing longitudinally or the transversely, seeds one or more suspended at the maturity by thread-like funicles. Just see the example. This is a Magnolia doltospora. Here you can say that there is the one thalamus is here. So thalamiflory. So number of stamens and the carpels are quite visible here. This is another species of the Magnolia hodgsonii. This is the Magnolia cathcartai. Here again you can see the number of stamens and the petals, and this torus is very visible here. And I said that thread-like funiculus are hanging here. So this is the thread-like. If you will see apparently, everybody will think that this is a thread, but this is not a thread. This is the funiculus. Another family, it is quite common in the northeast in India as well as the this tropical area in the plain. That's Annonaceae, where you will find the all three forms are here: trees, shrubs, or climber. But here no herbaceous plant. So if you the plant cannot be the Annonaceae. So likewise, you can apply your own that what characters were fitting. If it is fitting with the not fitting with that family, you just eliminate that family. So it will be helpful to you, and you are nearby to your family. Here the flowers usually are the bisexuals. Sepals here 3. Stamens are here again numerous, minute, filament short and thick, anthers opening by the longitudinal slit. Some of the anthers where you will find it is the dehiscing through the apical pore, like in Solanaceae. But in case of the Annonaceae, you will see that the opening by the longitudinal slits. Then carpels are the minute, usually numerous, superior. Then fruits carpels dry or fleshy. Here see the example. In case of Euheria Hamiltonii, here you can see the torus are visible here. Number of stamens, this yellow colors, and this central portion having the gynosium. Here Nymphaceae, the most of the plants are the aquatic. Easily you can identify. Then Caperaceae. In Caperaceae, there is the wide range of the habit. You will see that some of the trees are there, some of the shrubs are there, and some of the escandent shrubs are there, and herbs is also there. So if there is a herbaceous plant, this can be the Caperaceae, because you cannot directly eliminate this family because the wide range of the habit is here available here. Here you will see that flowers are the usually bisexual and rarely unisexual. So if flowers is the bisexual and unisexual, so you cannot eliminate this family. Likewise, you have to apply your own. Sepals here 4 to 5, petals 4 or absent. If it is absent, then it is the stixis. So you can see in case of the stixis where there is no petals. But another very important characters, there is one articulations on the peduncles, petioles, that is called the gynandrophore, where this mark is visible in fruit as well. So by these characters, you can easily distinguish this is family. In fact, only one character is sufficient to segregate the family, at least in case of the Caperaceae, where you will find that the ornamentations on the peduncles is present there. In Polygalaceae, here what is happening in Polygalaceae? This again comes under the thalamiflory. The sepal, petals, and stamens all are attached to the receptacle, and flower hypogynous, stamens many, and disc are here absent. Ovary 2 to 3 septate here. If you will see the fruit, here you can see there's very colorful arils are present there. So this is the Polygala arillata. Easily you can identify in the field also. Then the Caryophyllaceae, here there is no tree, only herbaceous elements you will find here. Here flowers are actinomorphic, sepals 4 to 5, stamens up to 10. Previous family having stamens more in comparison to the Caryophyllaceae. So Caryophyllaceae is the 22 number family according to the Bentham-Hooker system of classification. That means from Ranunculaceae to before the Caryophyllaceae, whatever the family is coming, their stamens are no more. Usually I am saying, not exceptional cases. Usually their stamens are more. Here in Caryophyllaceae up to 10, and these stamens will be the free here. Ovary superior, one-celled or rarely incompletely three-celled at the base, fruit a capsule opening by the apical tooth-like valves. In the Unknown-Aceae, you have seen that it is not opening through the apical pore, it is opening through a longitudinal slit. But here, it is opening through the apical tooth. Again, this is the one of the example. Brachysteमा calicinum, you can see this Caryophyllaceae plant. Then Malvaceae, it is a quite common family. From the +2 level, we are up to the MSc level. Every moment we are coming across with this family. So need not to go for the discussion. This is the Urena. Then this is the Abitulon. Then this up to this much was about the Thalamiflorae. Now come to the Disciflorae, where you will find the disc is present. See the example in the case of geranium. This is the geranium, where you will see that at the base of this there is a one disc. Disc is nothing; it is the additional layer which is giving the support to the flowering flowers. Means that means stamens, gynoeciums. In addition to the calyx, they are providing the support. And always remember, this will be always the below to the gynoecium or in support of or the in between. Then Rutaceae, very distinct family. Here you will see that there is the leaves, flowers and fruits usually wearing the numerous oil gland. How to know that? Just you crush the leaves, you will having the some smell. All the Lamiaceae, the Verbenaceae and some other plant also, Campanulaceae, they also having the some of the smells. But if you will see in the microscope, the clear glands will be visible on the fruit, leaves, flower in case of the Rutaceae. And again, there is the wide range of the habit here. It is not, there is nothing like that only herbaceous or the shrubby. Here you will find the herb to the tree. All kind of the things is available here. Flowers are the actinomorphic, unisexual or the bisexual. Calyx here 4 to 5 lobe, petals again 4 to 5 lobe. But fruit a capsule, clusters of follicles, berries or drupe. Like in Xanthogylum, you will see that it is a climbing shrub, where you will see that the cluster of follicles available there. But अगर आप उसमें माइक्रोस्कोप में अगर आप बाहर से भी देखते हो तो वहां पे आपको कुछ ग्लैंड्स दिखेंगे. If you will see the example of the Citrus medica, here you can see that this club-shaped stigma is here. And if you will see the means closely, the fruit having the some small this yellow greenish like structure. In fact, this is nothing. This is the gland. तो ये देख करके भी आप इसको पहचान सकते हो रूटेसी को. अनदर करेक्टर है कि उसमें स्पाइन या आर्म जिसको बोलते हैं, ये हो सकता है या नहीं हो सकता है. बोथ केसेस आर देयर. देन सेपेंडेसी, इन सेपेंडेसी लीची का फैमिली आपको पता है वहां पे एरिल होता है. बट अपार्ट फ्रॉम द लीची, जो भी स्पीसीज हमारा वाइल्ड में होता है, दे आर नॉट एक्सिबिटिंग द सेम करेक्टर. हियर यू विल सी दैट लोक्यूल्स, यहाँ जो ओवरी जो होता है ना, ये पेयर्स में आपको दिखेंगे. और जब मैच्योर हो जाता है तो समथिंग डिफरेंट, बोथ लोक्यूल्स बिकम द सेग्रीगेटेड लाइक दैट. और वो दो अलग अलग फ्रूट के जैसा दिखता है, केवल बेसल पोर्शन में दोनों अटैच्ड होता है. दिस इज वन एग्जांपल ऑफ द लेप्रासेंथिस सेनाग्लेंसिस. और इस पर्टिकुलर स्पीसीज में काफी वेरिएशन होते हैं. देन द कैल्सीफ्लोरी, हियर यू विल सी दैट स्टामेन फ्यूज टू द कैलिक्स ऑफ द फ्लावर. और इसमें जो मेजर ग्रुप है हमारा लेग्यूमिनोसी का, जो हम प्रैक्टिकल में भी आपको दिखाएंगे. सो यहाँ पे हम लोग 3 सब फैमिली में इसको डिवाइड करते हैं: सीजल पिनोइडी, माइमोसॉइडी एंड द पेप्लोइडी. सीजल पिनोइडी में कुछ नहीं, यू वी हैव टू सी दैट लीव्स इवनली पिन्नेट और द बाइपिन्नेट, समटाइम सिंपल और बाइलोब. इन केस ऑफ द बाहोनिया यू विल सी दैट द ऊपर में जो एपेक्स होता है उसका नॉस्ट होता है, वो आप पहचान सकते हो. एंड फ्लावर्स आर यूजुअली जाइगोमॉर्फिक. और इसमें जो होता है ना, स्टामेन्स की संख्या बड़ा इंटरेस्टिंग है, अप टू 10, दो यूजुअली 10 होता है. किसी किसी भी केस में, बाहोनिया के केस में जैसे आप देखोगे, there is the 2 species very similar to each other. One is the Bahonia variegata, another is the Bahonia purpurea. In case of the variegata, you will find out the number of stamen is the lesser than the Bahonia purpurea. उसमें 3 होता है, इसमें 5 होता है. So these are the some of the things you have to apply on. Mimosoidy, here you will see that leaves evenly bipinnate, rarely reduced to the, यहाँ पे bipinnate है और इसमें आपको लीव जो है ना सिंपल भी होता है. तो आप खुद से एनालाइज कर सकते हो कैसे इसमें डिफरेंसेस होता है. अनदर थिंग, In case of the mimosa, ये जो mimosa हम लोग देखते हैं ना, जिसको छुई मुई बोलते हैं, you will see that the inflorescence is looking very pinkish, and we are writing inflorescence pinkish. I must say that being a botanist, this is not the correct terminology, because the color of the inflorescence is coming from the stamens. जो exerted stamens होता है वो pinkish color होता है. Due to that they appear like that. But in general, the petals are whitish in color in case of the Mimosa pudica. Then papilionoides, here you will find that the stamens are 10 in and connect into a tube. Tube-like structure में होगा. It is a diadelphous. In case of Crotalaria, you will find that arrangement of the stamens are the 1+9. वो उसमें arrangement आप देखेंगे, जिसको हम लोग बोलेंगे diadelphous. Adelphous means together, means brother. दो अलग-अलग बंडल में जब रहेगा, तो that will be called as the diadelphous. But in case of Mimosoides, यहाँ पे stamen 10 to numerous होता है और वो exerted ज्यादा दिखाई पड़ेगा आपको. Like in case of Acacia catechu. ये देखिए आप बहुनिया variegata का flowers, और ये जो है again बहुनिया, पहले there was a Bahonia bigger family having the tree species as the lina, but as of now the world is accepting whatever the tree species, usually they comes under the Bahonia, and except this some of the genera species, usually I'm talking, and if it is a climbing, they will be transferred to the Phenera. ये आर्किडेंटोन में आप देखिए जो कि माइमोसोइडी फैमिली है, इसमें एक्सर्टेड ऐसा होता है जैसे कि माइमोसा में होता है. देन मैंने आपको पहले भी बता चुका हूं, लिप्स डिजिटेटली ट्राइफोलियोलेट एंड लिप्स पिनेटली ट्राइफोलियोलेट. In case of legumes, you can see that this is the one example where this leaf is the trifoliolate, and here you can see that pinnately I have wrongly written it is a trifoliate to confuse you. In fact, it is a trifoliolate. Remember terminology. Remember in past also you can see that trifoliate. It is not trifoliate. It is the trifoliolate, and here again see that trifoliolate. What is the differences? Here you can see that the petioles of this one is the longer than the rest of the two. So this will be comes under pedicly group. In case of the Saraka Asuka, very surprising, here no petals. Always remember some of the legumes where lack of the petals. This is the one of the example where you will find there is no petals. The colorful sepals are the only present here. The petals you will see that this is a scale like form here. Rosaceae, again very complex group. In Rosaceae, you have to depend upon the micro-morphological character. For that, again remember, very small and a precise description is needed for you. In case of the Melastomaceae, you will see here indumentum of the stellate, scale-like or the simple hairs. This plant is the either herbaceous or the shrubby elements. Leaves will be the opposite. Very beautiful family. Flowers bisexual, actinomorphic in cymes, panicles or clustered. Stamens are very important role. Stamens having very important role in distinct between the two different genera, like Osbeckia and the Melaustoma. Many having the doubt, but if you will see the number of stamens very carefully, you can distinguish between these two. Here stamens varies from 8 to the 10. Filaments are the geniculate. Geniculate means niche structure. जो कोई हमारा नी केहुनी जो होता है, वो नी के जैसा कुछ structures उस पे form करता है, जहाँ पे joint होता है. So that is called the geniculate. Anthers dehiscing by the pores. You see these photographs. Easily you can see this here. Small pores are available there, and through this the stamens, the anthers is coming out. Then begonia, it is the distinct family. Begonia में कोई tree species नहीं होता है, कोई climber नहीं होता है. Only this is the very small and weak herbs. और usually ये plant जो होता है जहाँ पे rock surfaces होते हैं वहाँ पे ज़्यादा ये grow करता है. इसमें flowers, you will see that the flowers are unisexual or bisexual or polygamo. So different forms are there. But male flowers में जो होगा perianth of the two outer valvate and two are the smaller inner segments. Stamens here you will find the numerous, free or connate below. Female flower perianth 2 to 5. Remember, in case of the male flower, the perianth numbers are lesser than the female flower. And here fruits a capsule. See the example of the begonia. Here you can see this, the capsule is like this one. In fact, this is a female flower. And this is the male flower. And this is the again male flower close up. And this is the fruit in case of the begonia. So this is a very diverse group. The plant, some of the herbaceous, all the looks like the herbaceous plants. This is appearing up to the 1.5 meter. In recently, some of the news I have seen that more than 3 meter begonia is also present in the world from some Thailand or some different part of the country, not from India. Cucurbitaceae, very distinct family. It is the climbing twiners, or you may call it as shrubs also, climbing shrubs like that. Then Hawksonia having this climbing shrubby because its stem is very hard. Then it may be the monoecious or the dioecious. Then calyx tubular or the campanulate, petals inserted on the margin of the calyx. Ovary inferior, carpels 3, usually 3. See the example, some of the species like appearing like this. Some of the species will be appear like this. And fruit having the lots of variation. So some of the fruits will be appear like this. Then coming to the gamopetally having the 3 series like this: inferiority, heteromere, and the bicarpellity. Then here in inferiority, ovary will be the inferior. There are the different orders. I am not going in details. Stamens, epipetalous, ovules, one to many. See the example in case of the Rubiaceae. Here Rubiaceae again herbaceous to the tree. So wide range of the habit. But remember, this is the 84 number of the family. In previous family, you have seen that petals are free. So once the petals are free, you have to only look into the 83 family. Don't look into the above the 83 family. For that, you have to remember the synoptical key of the Bentham-Hooker system of classification. This cannot be, no other can make you memorize. You have to do your own. If you are knowing the family sequences in Bentham-Hooker system of classification, your half part is done. So at least you are knowing which family will be fitting where. So this is the importance of the Bentham-Hooker system of classification. Rubiaceae having the gamopatly. Here you will see that herbaceous to the tree. But since the tube is the fused, so you need not to check the some other place, other family. Here this is the Cophia. This is the Mitragana. Then Musenda. Musenda is the entirely distinct plant in the forest. How it is? If you will see that sepaloid, this is the very only one calyx is enlarged. This one enlarged calyx, and that it is useful into the attracting the insects for the pollination. So by this you can easily identify the Musenda. Musenda having more than 50 species in India, and most of them having these characteristic features. If it is not present, so you can key out like that, the one in calyx enlarged, calyx not enlarged. So likewise you can make the two distinct group. So in inferiority, this Asteraceae, and in Asteraceae there is a wide range of the flowers. Here easily you can identify to see the flower head. In fact, this is the inflorescence. Don't say it is the flower, it is the inflorescence. So some of the unusual flower, like in case of the Elepenthupus escaver. Then Ericaceae. Most of, usually in the case of Northeast India, if you are visiting any forest, you will come across, encounter with this particular family in case of the rainy season, summer season usually. Like again, they having the wide range of the habit, like it's starting from the epiphytic to the shrub and tree, but no herbaceous plants are there in case. Here you will see that the corolla, petals united at the base and almost to the apex, tubular, barrel shaped, urn-shaped, campanulate, funnel shaped. So lots of variations in the case of corolla you will find. Number of stamens are here 5, 10, 18. Up to the 18 they are going. And thus opening by the apical pores, sometimes spurred. Fruits again vary or 5 to 20 valve. This is the one of the example of the rhododendron. Since I have very limited time, just I am moving very fast. I am just going for the pictorial pictures only I am showing. This is the Plumbaginaceae, where you can identify through this glandular hair. In Styrax, you can identify through the, here stamens are opposite to the petals. So that is why heteromere, ovary here superior, stamens are the opposite to the petals and double the petal numbers. Then the bicarpellity, here ovary superior with the 2 carpels. This is the example of the rhytia. Here you can see that is the conifant of the stamens are there. It is visible from the outside. So most of the rhytia having these features. If it is the Asclepidaceae, you can find out the some pollinia. Here is every angle having the some pollinia. So presence of pollinia is the characteristic features of the Asclepidaceae. Now question comes whether we should follow the Apocynaceae and the Asclepidaceae. I must say that if you are following this APG system of classification, you have to write the Apocynaceae. If you are following the classical system of classification, you have to follow the Asclepidaceae. But this is not wrong. So in my personal opinion, I feel that Asclepidaceae should be a distinct family. Again, if you have the fruiting materials, how to identify the Apocynaceae and the Asclepidaceae? If the needle shape seeds are there, that will be belong to the Apocynaceae. If there is the some ovate or the some different structures of seed are there, that means it is Asclepidaceae. So this is my personal observation. In most of the cases, I have noticed that this is the very correct character, in fact. In Solanaceae, there are a range of the variations from herbaceous to the tree species. You can see that in the case of the Solanaceae. Then in case of Solanum, you will find out that is the apical pore is present there. So apical pore in previous, if you see the case of Annonaceae, there was some longitudinal silks are there, but here again this apical pore are the present, which is the characteristic features of the Solanum genus I am talking. Then Scrophulariaceae, again in Scrophulariaceae, most of the species are the herbaceous. Some of the shrubs or tree is also present, but it is quite rare. And usually you may find this species everywhere. Not like that this is the for hilly region or the ground plain area, but everywhere this family is available. Then Acanthaceae, again this is a very wide range of the family. In past, the Thunbergiaceae was the one family because of the climbing nature, but at present it will comes under the only Acanthaceae. This is the Astrobilanthes. You can see that this is a climbing and this is a erect shrubs. So this is the basic difference, means a wide range of the habit in case of the Acanthaceae. Lamiaceae, again here the zygomorphic flower, and in Lamiaceae, the only tree species in entire India is the Leucoseptum cannum. Then this is the Anisocylus carnosus. Then Hyptis subulens easily can be identified. And this is the Gomphoistema henianum. I mean, this particular species is growing in the southern India also, on the northeast India also. So don't confuse that southern India plants is different than the northeast India. Both are the henianum only. Phogostemon auricularius. Then we are coming to the monoclemides. Here you will find that this sepal petals have been fused and only one perianth is forming here. This division was never intended to be natural. It was merely to be help identify these single perianth plants. Some are the basal families, Aristolochia, Piper, with unusual flower structures, whilst others are highly derived with sepal and petal lost. Engler and others appreciated this soon after the publication of Darwin's Origin of the Species. So this is not a natural group. You will find out lots of variations are here. In the case of Nyctaginaceae, you may see that the stamens are exerted. Some of the, in usually cases, it is not exerted. In case of Polygonaceae, you will come across with two genera, this Polygonum and the Persicaria. So basic differences between the Persicaria and Polygonum, you need not to go for the inflorescence and other characters. Only you have to see the ochre. If you are knowing the ochre characters, you can easily identify it. Say for example, if you will see this ochre in case of the Polygonum, here you can see this ochre. It is segmented like this structure. So this will become under the Polygonum. And if it is not segregated, then it will become under the Persicaria. So this is the basic differences between the Polygonum and the Persicaria. You need to check the ochre very carefully. no other characters are important than the ochrea. So this is the distinct family, no need not to go on the details. So this is the flower. Aristolochia. So this is also unique family. Easily you can identify in the field. Today we will see this in practical classes also, this Aristolochia. Laudaceae, again I said that this is again wide range of the some of the climbing. Cassitha filiformis is the one genus which is having the climbing nature, not growing in the northeast India, usually growing in the Odisha, Bihar, Jharkhand. I have seen this one. So there again, this habit is the quite diverse. So you have to check all the plants very carefully. I must say that this is the one very unusual family. In fact, in Elaeagnus you will see that some scales are present over the stems and the leaf beneath, and easily you can identify with the silvery scales. This particular plant is growing in the hilly area, and you can see this plant from the distance. You may say that this is belongs to the Elaeagnus. Laurentheaceae, again it will be most of them are the epiphytic, and from the distance you can identify this particular plant. Euphorbiaceae, very beginning, from the beginning you are knowing that there is a one cup-like Scythia, unisexual actinomorphic, and the usually plants are the monoecious or the dioecious, and most of the flowers are the unisexual. Seed may be the arillate or not arillate. Here you can see that trilocular ovary is here present there. So by that you can identify this is some Euphorbiaceae plant. And this Scythia inflorescence is here, Pulcherrima. Then Ricinus, it having something more than 100 stamens are there. So this is a quite interesting plant. The flowers, male flowers are the downside, and the in apical region you will find the female flowers. Phyllantheraceae is the distinct than the Euphorbiaceae due to that position of the flowers. Usually the flowers are in axil of the leaf, and it is the lower side. So that is why it comes under the of Phyllantheraceae. So distinct family. Moraceae, everybody is knowing this ficus, so need not to go in the details on that. Then Salicaceae, here the plant is the dioecious or the rarely bisexual tree and shrubs. Here you can see that catkins, often seriaceous or villous, at least first terminal leaf sessile, lateral shoots or sessile axillary. Then discs are flat. Male flowers with the 2 to many stamens, filaments free or united. Female flowers with ovary solitary, sessile or shortly stipated, one locular. Capsules ovoid 2 to 4 valved. Seeds few to numerous. See the one example in case of the Salix tetrasperma. Here the seeds are the 4. Easily you can identify in the field. Usually grows along the river side. So this much, and monocotyledon is the distinct in the from the dicotyledon. Here no complexity in the case of monocotyledon like the dicotyledon. So this is all about my presentation. So Abhishek sir, this is over to you now.
Thank you, Dr. Gopal. Now we will start the practical part. And Dr. Anand will start with some plant of Gamopetali. So, welcome, Dr. Anand.
Yeah, well, good afternoon to all of you. Just today, you all have learned about plant identifications from various lectures. Now, I am going to present a practical approach on how to identify the plant. Thus, I am focused on the Gamopetali. You all have learned. You all have learned there are 3 classes: polypetally, Gamopetali, and monoclinidae. Okay, and the polypetally means petals are free, and Gamopetali means petals are fused. So I'm focused on only the Gamopetali where petals are fused. Now this is I have this plant. So how to characterize this plant? First of all, we have to write down first of all habit of the plant. What is the habit? Herbs, shrubs, climbers, or tree. This is the tree plant. So we have to write the tree, and you have to write down the height of the plant. What is the height of the tree? Then in case of the tree, you have to also note down about the bark character. What is the bark character? If the bark is peeling, so you write down the bark is peeling. If the bark is fissured, then you write down the bark fissured. If the also write down on the color of the bars. And the color of the bar is the white, either white or brownish, any color you have to write down. So this is the tree species, and this is the tree. Then after the noting down of the bar character and the height of the tree, just you go to the stem. What is the nature of the stem? If the stems are round, means straight, then you write down the stems are straight. If the stems are angled, just you see these stems, there are some showing the angles. So you write down the stems are the angled, and then see the nature, hairy nature. If they are glabrous, then you write the glabrous. If there are some hairs, then you write down the pubescent hairs. Then after the stem character, just you go down to the leaf character, the orientation of the leaf. Here you can see the leaves are the opposite. The leaves are the opposite, and then you write down the leaves are the opposite. If they're simple or compound, then you write down the leaves are the simple. Here the leaves are simple, opposite. Then you go down to the whether the sessile or petiolate. There is nothing is the petiole, so you write down the sessile. Then go from the shape of the leaf. What is the shape of the leaf? Then size of the leaf. Then what is the nature of the base? Then margin. Then apex, and then the texture. What is the, if the glabrous, you write down the glabrous upper surface. If the pubescent, write down the pubescent. This is the upper surface, and this go to the lower surface. Then you have to also note down the veins, secondary veins. Then what is the number of the secondary veins? After the leaf character, just here you can see, you see this. There is one stipule. This is not an interpetular stipule is present here. After characterization of the leaf, then we have to go for the inflorescence, because the flowers and fruits are the most important things for the identification of the plant. If we are not having the flowers or fruits, then it's very difficult to identify the plant. So here we are seeing that, just you see, this is the inflorescence. This is the inflorescence, okay, and this is the peduncle. So the inflorescence is peduncle inflorescence is present. And what is the position? This is the axils of the leaf. So the inflorescence are the axillary and peduncle. And what is the type of the inflorescence? Just you see, this is the head, peduncle. This is the peduncle head. Means inflorescence is the peduncle heads, flowers in peduncle heads. Yeah, just you see, this is the axils, the inflorescence is axillary, and flowers in the, this is the peduncle head, this is the heads, peduncle heads. Okay, then we have to see, yeah, just you see that this type of structure. Actually, this type of structure is the enlargement of the hypanthea. Hypanthea actually enlargement of the base of the structure of the base of the calyx. This is the, actually this is the typical character. This is the typical character of the genus Morinda. Okay, so we have to write down the flowers. Flowers are attached by the enlarged hypanthea. Okay, then you just see the calyx. Calyx are also the tubular, but here the lobes are obsolete. Then we go for the corolla. Okay, then this is the tubular structure. So the corolla is the tubular, and this is the tube. So you have to write down the what is the length of the tube. Then we go for the lobes. Just you see. Just you see. You write down the, first of all, number of the lobes. 1. Here. 1, 2, 3, 4, and 5. This is the numbers of lobes. Then you have to dissect. You have to dissect the flowers first of all. Just you dissect. Yeah. Just you can see the number of stamens. You can see 1, 2, 3, 4, 5. And then stamens number is the 5. Then you have to measure length of the filaments and the anthers. You have to also, if there is any color of the anther, black, brown, red, then you write down the color of the anther. Just you dissect this also tube. Yeah. Okay. Then we go for the fruiting structure. Just you see this. This is the fruiting structure. This is the fruiting structure. This is the mass of the that's the hyphonthia, and this is the peduncle. So after characterization, you see this interpedular stipule. Okay, actually the interpedular. If you are finding the interpedular stipule in any plant, then directly you go for the family the Rubiaceae, because the interpedular stipule is the typical character of the family Rubiaceae. So this plant belongs to the member of the family Rubiaceae. Then earlier I told also that the flowers are connected through the enlarged hyphonthia, and this is the typical character of the Morinda. So the plant belongs to the genus Morinda. Now we can go for the species. What is the species of the plants? What the nature of the leaf pubescent? Whether the leaf is if the leaf is a pubescent, then it is a tomentosa. If the glabrous, then the other things. So this is for the Rubiaceae element for the Morinda. Now I'm going to the other plants. This is the plant of the other group. The other group is the Asteraceae. In the Asteraceae, the flowers in the always in the head form, and the flowers in the heads, and all the florets are enclosed in the involucral bracts. And in Asteraceae, the most important thing, achins are the fruit types of the Asteraceae members, and it is one of the important character of the identifying the species belonging to the family Asteraceae. In the Asteraceae, there are two types of inflorescence. One is the discoid inflorescence, and one is the radiate inflorescence. You can see if there are single type of the florets. Florets is the unit of the flowers of the inflorescence of the member of the family Asteraceae. Family is Asteraceae. Actually, discoid flowers are inner part of the inflorescence, and radiate flowers just are the ligulate flowers. Oh, these plants belong to the Sonchus, and you see the leaf also. This is the leaf nature. This is the amplexicol leaf. Okay, this clasping the whole leaf base, clasping the stems. So these plants belongs to also the Sonchus oleraceus. So in the case of the Asteraceae, achins are the most important. Now I'm showing the another group, and so you can also separate the Asteraceae plant and Rubiaceae plant. Just you see here the interpedular stipule present in the Rubiaceae plant, where in Asteraceae no interpedular stipule present. you can see here achenes are present, here not achenes are present. In Asteraceae, you can see the in Asteraceae calyx lobe reduced to the pappus, hairiness structure is pappus. But here in Rubiaceae, calyx lobes are distinct or obsolete. That this is the major difference between the Rubiaceae plant and the Asteraceae plant. And both plant having inferior ovary. Now I am showing the another group where the ovary are the superior. Just you see this plant, okay. Here the juice is milky. If you just break the stem, then there is a one juice, milky juice is present. Just you see, this is the stem, this is the lenticels are present, means stems are the lenticulates. Just and leaflets are the opposites, okay. Then in and just you see the flowers. In flowers, here you can find the corona scales. This is the corona scales, and just you see this is the anthers. Anthers convenient means rounding the rounding the stigma, okay. And this is the typical character of the ratia. This is the typical character of the ratia, where all anthers means five anthers are connivent round the stigma. And this is also here you can find the corona scales, and this is the lobes. So this is the typical character of the ratia. And under the ratia there are many species, just here arborea, coccinea. And the coccinea is the reddish color flower, and this is the creamish color. The lobes, corolla lobes are the creamy color. So this plant belong to the Ratia arborea. And just related, you see, this is a one. This is another Apocynaceae member. Just this is a very common plant. This is the Rolfia. Okay. This is the Rolfia serpentina, where you see the leaves are the, trinately, leaves are in the three, trinately arranged. And the inflorescence also is the, just you see the flowers, just you see, calyx are also the lobed, corolla is the tubular, okay, petals are 5-lobed and overlapping. Here the petals are also overlapping to the left. So this is the Rolfia serpentina, and this is the one is the closely related group is the Asclepiadaceae. Yes, just you see the Asclepiadaceae plant. This is the climber in nature, and here also the milky juice is present. But how to differentiate between this plant and this plant? Just you see this flower. If you open this flower, if you open this flower, here you can find the corona, okay, where you can find, this is a very teeny parts. So yeah, this is very small. This is the corona. There is the corona scales present, but here is the corona. This is the major difference between this plant and this plant. And this plant belongs to the Tylophora indica, where the leaves are also opposite. Flowers, you see the peduncle. Here is the peduncles, and this is the umbellate signs. Okay, this is the inflorescence type. So this is the major difference. There is the corona scales present, but here is the corona present. This is the climbers, and this is the tree species. So this is the Asclepiadaceae I have shown, and Apocynaceae. And now I am showing the another group. This is the Solanaceae member I am showing. I am showing the Solanaceae member, this plant. Just you see, this is the one minute. This is the Solanaceae member. But you see the This is the fruiting structure. This is the 1, 2, 3 tricot. This is the signs. Okay, you can see trichotomously branched signs. And this is the one thing. This you see. This is the armed structure. Here is this is also the one arm. In the Solanaceae, there are two. One plant is the arm, and one plant is the unarmed. If you find the Solanaceae member, then you can also key out between the armed. One plant is the one group, armed plant, and another group is the unarmed. Here you can segregate. This is the armed plant. Okay, and just if you see the flower, just you see where is the flower. Yeah. This is the flower. Flower color is the white color in nature. And in the Solanaceae, actually the stamens color are here. Stamens color are yellow, but the length of the stamens are more than the length of the filament, and also dehiscence from the pore, apical pore, not from their length. So the dehiscence from the apical pore and the length of the anther are longer than the filament. This is also the typical character of the solanum. So under the solanum, we have also differentiate between the armed plants and unarmed. This is the unarmed plants. Okay. Now just we see the leaves, where this is the leaves. The base of the leaf is the unarmed. There are some plants where there are spines are present. Okay, here there are no spines are present, and this is also the typical character of Solanum torvum. So we can identify this plant. Our plant belong to the Solanum torvum. Our plants belong to the Solanum torvum. Just you see the another group, under the Solanaceae, the same family. Just you see the Solanum diphyllum. Why it is known as a diphyllum? Just you see, yeah, Solanum diphyllum. Okay, just you can see at every node 2 leaves arise. Okay, 1, 2, 1, 2, 1, 2. 1, 2, 1, 2. So this is also the typical character of this species, is Solanum diphyllum, whereas the fruit color is the yellow. So we can identify that it belongs to the Solanum diphyllum. And another member of the Solanaceae. Okay, just you see, first you see the fruit. So this fruit. This fruit is the capsular. Okay, under the Solanaceae, this is the capsular, and you see this fruit means Solanum diphyllum fruit is the berry. So this is the major difference between the two genera. This is the Solanum, and this is the Nicotiana. In Nicotiana, the fruit is the capsular, while in the Solanum, the fruit is the berry. This is the major difference between the Solanum and Nicotiana. You can easily differentiate between which one is the Solanum, which one is the Nicotiana. And Nicotiana is also the inflorescence is the terminal panicles, either terminal panicles or racemes. This is the I'm sorry, I think this. Yeah. Now I am showing the members of the. So these all plants have regular flowers. Okay. Now I am showing the irregular flowers where the corolla lobes are the bilobed or the obliquely bilobed. So I am just showing the first family is the Acanthacea. Okay. I am showing Acanthacea. Just you see the Acanthacea member. Okay. In Acanthacea, where you will also find the leaves are the opposite. In Acanthacea, reticula. Okay. If you reticula is the structure, actually the seeds. The seeds are supported on a small bony type of structure that is known as the reticula, and reticula is the important diagnostic character of the member of the Acanthacea. But there are some exceptions where reticula is absent, like the Thunbergia and the ammonia, where the retinicula is absent. But most of the acanthus members of the plant having the retinicula. Just I'm showing this plant. Okay, this is the Ruilia. Okay, where is the flower? Actually, the flowers are wilted down. Where is the flowers of Ruilia? No flower. Showing just 1 minute. No, I am not taking the flower. Okay, I'm telling about the flowers. Actually, there's a Ruilia having the 4 stamens, and the stamens are the diadynamous in the nature, means 2 small and 2 large. Okay, and just you see, bracts are also the important characters, where some of the bracts are leafy and some bracts are the linear. Where you can find, you see this is the bracts, where this is not the leafy, the small teeny structure, and just you see the calyx. Calyx are also this is the fruiting structure. The calyx are the 5. Okay, the 5 lobes, and lobes are the just linear. And just you see the fruit shape. The fruit shape is the linear oblong. The linear oblong. The fruits are the linear oblong. And just I'm showing the another plant of the same genus, just where the fruits, another group. I think the fruits are fallen, so just leave it. Here is the fruits are the here is the fruits are the just you see the linear oblong. So this is the also the character of the Ruilia tuberosa. Okay, and if you just see the one is the also the Ruilia prostata, where you just when you see the fruits, the clavate nature, clavate, the fruit shape is the clavate. If you find the fruits are the clavate in shape, then maybe it's the Ruilia prostata. And in the Ruilia prostata, or the one major important or diagnostic criteria is the bracts. There is the bracts are the leafy. If you find the bracts are leafy in the Ruilia, just a closely species means Ruilia tuberosa and the Ruilia prostata, then it's belong to the Ruilia prostata, and this is not leafy, this is the Ruilia tuberosa. Then you can easily distinguish between the 2 species under the same genus. Now this is the acanthus member, and also one important character is the Thunbergia. All are the acanthus member are the herbaceous or shrubby nature and climbers, but the one not climber, but only the Thunbergia is the climber in nature. The another plant of the acanthus member, just you see, this is the climbing. This is the climbing in nature. Okay, and the flower is the creamy in color, and just you see the inflorescence. You see, this is the only one side. You can find the one side flower, not in this side, only one side, and the flowers are pedicellate. Okay, so this is the terminal racemes. Terminal racemes, and we can call terminal one-sided racemes, not two-sided, one-sided, because all the flowers are arranged on one side. Okay, so the inflorescence is the terminal one-sided inflorescence. And so this plant also, it's the fruits are the capsular. Okay, and this plant is the Acacia gangetica. Leaves are also the opposite and climbing in nature. So this is all for the acanthus member. And just you see this plant. This plant belongs to the Lamiaceae member. For all the Lamiaceae member, if you pluck the leaves, this is the leaves. If you crush this type, there is some scent. there is sand. So just you go for the Lamiaceae member. This type of there is small sand, aromatic. This is aromatic in nature. Okay, so easily you can go for the family is the Lamiaceae members, and the flower actually this is the fasciculed flowers are the arranged in the node, and the flowers flowers are the bilabiate, whereas the upper where is the this is the lower lobe, and stamens are arranged two stamens are these stamens are arranged here and upper lobe and the lower lobe also. So this plant very hairy in nature, and the N. isomelis. This plant belong to the N. isomelis, and this is the member of the Lamiaceae. So another member is another member of the Verbenaceae. Okay, where is the fruit in nature in the drupaceous nature. Fruits are the drupaceous nature. The fruits are drupaceous in nature, and then just you see the inflorescence. Here, inflorescence are axillary, and just you see the peduncle, and it is in the head. Okay, in this plant very hairy in nature, and this is a lepia. There are two types of plant. One is the erect in nature, and another is the prostrated nature. That one, the erect, where the erect in nature, that is the lepia alba, and the one is the prostate in that one is the prostate in nature. This is the lepia nodiflora, that is also known as the phylum nodiflora. So you can easily distinguish the species under the same easily. These types of characters you have to identify. So that's all from my sides under the gamopetly. Okay, now Dr. Vijay Masavkar will explain about the polypetly group, where the petals are the free. Okay, thank you.
गुड आफ्टरनून ऑल ऑफ यू, नमस्कार। जैसे कि हम जानते हैं कि डॉक्टर गोपाल कृष्णा सर ने इतना अच्छा प्रेजेंटेशन दे दिया, सब कुछ उसमें कवर हो चुका है, लेकिन उसका हम कुछ लाइव करेक्शन किया है गार्डन से, उसके बारे में थोड़ा बता देंगे। जिसमें ये पेप्लोनोइडी के बारे में बताएंगे कि कैसे क्रोटोलेरिया पेलेडा, जिसमें ये ब्रैक्ट्स के आधार पे और इसके फ्रूट्स के आधार पे हम लोग इसको पहचान सकते हैं। इसमें ब्रैक्ट्स होते हैं और इस टाइप का फ्रूट्स होता है। इसी प्रकार से ये जाइगोमॉर्फिक फ्लावर्स होते हैं इसमें, और इसके अलावा ये माइमोजेसी का एक प्लांट है जिसमें कि फ्लावर्स एक्टिनोमॉर्फिक, कैलिक्स एंड कोरोला, वेलवेट इन बड, माइमोजेसी, इन्फ्लोरेसेंस इन स्पाइक्स और आल्सो इन ग्लोबोस हेड में भी होता है। साथ ही इसमें थॉर्न्स होते हैं माइमोजेसी में, और कुछ में नहीं होते हैं। इसमें दो डिस्टिंक्ट जेनेरा आल्सो डिस्टिंक्ट किए हैं, थॉर्न्स के बेस पे और विदाउट थॉर्न्स के बेस पे, जिसको सेनेगेलिया और बेचेलिया जीनस भी कहा जाता है। और इसमें स्पाइक्स एक्टिनोमॉर्फिक फ्लावर्स होते हैं और इसका फ्लैटेंड फ्रूट्स होते हैं। दिस इज द फ्रूट्स। सीजल पिनेसी ग्रुप में अभी दिखाया था गोपाल कृष्णा सर ने कि इन्फ्लोरेसेंस में ये सराका अशोका कलेक्टेड किया है, जिसमें कि ये पेटल्स इसमें एब्सेंट होते हैं। सेपल्स ये जो कलरफुल दिख रहे हैं इसमें अपोजिट इसके ब्रैक्ट्स होते हैं, और इसमें ब्रैक्टियोल्स के आधार पर इसको स्पीसीज में डिफरेंशिएट किया जाता है। और इसमें लीफ प्लेट्स कंपाउंड है, और जो कि इन पेरिपिन्नेट और पेरिपिन्नेट के बारे में बताया गया है। ये पेरिपिन्नेट लीफलेट्स आप देख सकते हैं, और इसका फ्रूट नाम से और ये इसका पोड है। लेग्यूम्स में पोड का बहुत इंपॉर्टेंस है, जिसमें कि इसको लोमेट्स और पोड्स और लेग्यूम्स कहा जाता है। जो कि सराका अशोका का पोड, ये आप देख सकते हैं, इस टाइप से आप इसको आइडेंटिफाई कर सकते हैं और देख सकते हैं कि ये सराका अशोका का पोड है। फ्लावर जाइगोमॉर्फिक और पेरियंथ सेगमेंट प्रीडोमिनेंटली इम्ब्रिकेट इन बर्ड्स में होता है, सिजलपिनेसी और पेप्लोनाइडी में, जबकि दोनों सब फैमिलीज को हम डिस्टिंक्ट कर सकते हैं। कोरोला सिजलपिनेसी एस्टिवेशन इम्ब्रिकेट्स असेंडिंग पोस्टीरियर पेटल्स, इनरमोस्ट द पेटल्स टिपिकली 5 एंड डिस्टिंक्ट होता है सिजलपिनेसी में, और कोरोला पेप्लोनेस एस्टिवेशन इम्ब्रिकेट डिसेंडिंग इन द पोस्टीरियर पेटल्स, outermost the 2 anterior petals forming the kill, जो किल बनाते हैं, जो कि पेप्लोनेसी में होता है। तो ये हमने केवल लेग्यूमिनेसी का देखा है। अब नेक्स्ट ओवर टू डॉक्टर श्याम विश्वा।
Good afternoon, everybody. So now I'll be dealing with some of the families. So you can just have a look of this particular plant specimen. So you can see the particular plant in this. Are you able to observe this particular plant? So when I break it, and you can see the white latex coming out from this. The white latex, characteristic feature of this particular family. And when you look at this, the flower, there is no distinguishing between petals and sepals. So here you can say the flower is having the perianth. So it's a characteristic feature of this particular family known as Euphorbiaceae. So the characteristic feature of the family Euphorbiaceae is that most of the plant you will find it is having the latex in it. There are many specimens or species are there which are having characteristic feature in which there is, if you observe the fruit, there is trilobate structure. You must have eaten amla. That also belonging to the family Euphorbiaceae. So here in this particular genus Euphorbia, it is having thorny structure in it. So you may be confused with when you look at the thorns, it looks like cactaceae. But looking at the flower, the other characteristic feature, fruit, you can easily come to the conclusion that this particular family belongs to the family Euphorbiaceae. And this particular genus is Euphorbia. When you observe the flower, there is a characteristic feature, inflorescence, which is known as cythium. Okay, so there is in this cythium, there are two flowers in it, the female flower as well as the male flower. The male flower is surrounding the female flower. Okay. So one more, it is here, this particular plant specimen, just have a look. So this is a herbaceous prostrate plant. It is a herbaceous plant. So you can see a minute flower at the node, it is present. So here also it is having the inflorescence, that is cythium is present out here. Okay, minute flowers are there. So when you'll observe under the microscope, you can easily make it out what are the how the male flower and how is the female flower looks like. So though it is not maybe visible to you all, but if you observe under the simple microscope, the characteristic feature or the how the inflorescence is when it is there, you can easily make it out and you can come to the conclusion that it is belonging to the family Euphorbiaceae. And this is Euphorbia hilta. Okay. So one more near to that, this, you can see easily, you can make it out. This family belongs to the nettle family, or you must be aware of this family. It is known as Sishnu in Sikkim and all. People are the leaves are being eaten. So you can see in this the leaves are stinging in nature. When we were kids, we used to sometimes they used to have a good sort of thrashing with these leaves. Okay, so it is having stinging in nature. The leaves easily people don't touch with the leaves, don't touch with the hands, because the leaves are very having stinging nature. Okay, so this belongs to the family Articaceae. So here one more family is there. Our botanical garden is very famous because of this particular family. So here when you see, one of the students asked me, where is the flower? Okay, looks like a fruit, but this family is characteristic feature of this family is the fig. This is the fig, it is known as. So when you look at the fig, when you cut open this fig, so just have a look. When you dissect it, when you open it, it is having many flowers in it. So there are male flowers, the female flowers, the neutral flower, and the gall flower in it. When you observe under the microscope, you cut open. So there is a special characteristic feature known as the hypanthoddium. The inflorescence is known as hypanthoddium. When you open it, you will see the different flowers in it. It is known as the male flower, the female flower, the gall flower may be there, or maybe the neutral flower will also be present here. Okay. So there are here also you when you observe, when you a milky white latex may be present in this family also, belonging to family Moraceae. Okay, when you observe the leaves, sometimes when you observe the leaf, you can see here there is, you can see here the leaf, it is having three from the base, it is having three nerves, 1, 2, and 3. You can observe the 3 nerves can be visible in this particular leaf. And one more, I'll show you. So here, one more Moraceae family is there. So you can observe at the back side also, if you are not able to count, look at the back side. So 1, 2, 3, 4, and 5. So 5 nerves are at the base. So based on this characteristic feature, you can differentiate between these two plant. This is also a tree, this is also a tree. But when you look, this is also belongs to Moraceae, this is also belongs to Moraceae. But when you observe the venation pattern, so you can easily make it out. It is having 3 nerves. It belongs to the genus Ficus altissima. But this particular genus, it is having 5 nerves at the base. It is Ficus bengalensis. Okay, when you if you visit botanical garden, our famous the Ficus bengalensis, you can see the banyan tree in our garden. Okay, so one more family is here. This also is so morning we had collected, now it is already dried up. This also belongs to the family Moraceae only. So but the leaves are a different shapes and size. That's why you can see the fig, it is attached out here. So this is the fig, and this is a herbaceous plant. You can say this is a straggler, or it is a creeper plant. It is also belongs to family Moraceae, Ficus heterophyllus. Okay. So you can see one more, a very large one. So this is a fruit, edible fruit belonging to Moraceae only, but the genus is different. Okay, so it is a monoecious plant or dioecious also. So you can see the Moraceae, just now I had shown you the ficus. There you will find the both the male plant, female plant in the fig or in the hyponturium. But here you will find a different structure. This is the female part and the male part will be in a different plant. Okay. So here you must have seen that cuttle, ficus, jackfruit. The common name is jackfruit. I am not able to recall it. So the jackfruit, a very large fruit, which is present at the trunk. Okay. So that belongs to the Artocarpus heterophyllus. So this is Artocarpus. This is a different Gomesiana. So here, when you observe this plant, so from this plant you can make it out this is a climber in nature. Okay, so it is having somewhat this tendrils and all. So these are some of the field characteristic feature which you will have to observe it. And you can see here the flower, it is there. So when you take one flower and you see the count the number of perianth. So it is having 1, 2, 3, 4, 5. 5 or 6 perianth is present. The stamens is there, and you if you observe the leaf characteristic feature, it is having somewhat you can see spade-like structure is there. The base is cordate. So these are some of the characteristic feature which you will be observing under the microscope, and belongs to family Polygonaceae, and the genus is Antigonan leptopus. Okay, so just now you have been told that belonging to between Polygonum and Persicaria, that is the awkward stipule, it is present in the base of the leaf. When you observe it, so easily you can make it out. So the family Polygonaceae. So one more family is there. So you can see this particular plant specimen. So from the habit only you can make it out. This particular plant is a climber, climbing in nature. You can see, and when you'll observe the flower here, so you can see the perianth. It is modified into tubular structure. Okay, when you open this tubular structure, you will see the inside of which both are both the stamens and pistil. It is conjoined, or you can say that it is forming a gynostragium structure. It is forming, and when it is matured, it is forming a capsule. This it will form a capsule in structures. So this is some of the this is a characteristic feature of the family Aristolaceae, and this plant specimen belongs to the genus Aristolochia. Okay. So next, I'll transfer this one to Dr. Gopal. He will come to the monocot section.
वो ये बाकी स्पेसिमेन था ना Okay. Basically, in my talk, I mentioned about the many things like how to classify the plants. So first job is our classification. So in grouping of the plants, what we will do? in grouping of the plants, we will group in the two. Firstly, it is the either angiosperm of the gymnosperm. Here we don't have any gymnosperm. Therefore, simply we will talk about the angiosperm. In angiosperm, again there is the two group. One is called the dicotyledon, another is called the monocotyledon. अभी डाइकॉट और मोनोकॉट में मैंने डिफरेंशिएशन बताया था, उसमें कोई वो नहीं है. ये अब डाइकॉट है या मोनोकॉट है, ऑब्वियसली दिस इज द मोनोकॉट. इससे पहले जितने भी प्लांट दिखाए थे आपको, जितने भी दिखाए थे उसके और इसके लीफ में भी आप डिफरेंशिएशन अगर देख सकते हो, एक्सेप्ट सम ऑफ द स्पीशीज वेयर डाइकॉट इज शोइंग द मोनोकॉट कैरेक्टर एंड मोनोकॉट इज शोइंग द डाइकॉट कैरेक्टर. उसको छोड़ के सभी में अगर आप देखोगे, वन हैविंग द लीनियर लीव्स एंड द पैरेलल वेनेशन, अनदर हैविंग द रेटिकुलेट वेनेशन. तो इन दोनों को देख करके आप इजीली आइडेंटिफाई कर सकते हो कि वेदर दिस इज द मोनोकॉट और द डाइकॉट. तो ये मोनोकॉट में है. मोनोकॉट में बहुत सारी फैमिलीज आपको मिलेंगे. 166 के बाद जितनी भी फैमिली बेंथम हुकर सिस्टम ऑफ क्लासिफिकेशन के हिसाब से, दीज ऑल आर बिलोंगिंग टू द मोनोकॉटिलिडॉन. मोनोकॉटिलिडॉन्स में ये हमारा सबसे एक डिस्टिंक्ट फैमिली है. इसको आप कहीं पे भी जंगल में जाओ या कहीं रोड साइड में मूव करो, आपको मिलेंगे ही मिलेंगे. और इसको पहचानने के लिए बेसिक तरीका क्या है कि इसका सबसे पहले आपको ये देखना है, ये जो नीचे का जड़ है तो होता है ना, ये कभी कभी टफ्टेड होता है, काफी ज्यादा ये होगा, मतलब उसको आप इजीली आप नहीं रूट आउट कर सकते, उसको निकाल नहीं सकते. तो इससे आपको समझ में आ जाएगा कि वेदर दिस इज द एनुअल और द पेरेनियल. और जो इजी आ जाएगा वो एनुअल्स है. प्लस इसके पास में यहाँ आपको ये कुछ स्ट्रक्चर्स भी आपको दिखेंगे जिससे आप इसको पहचान सकते हो, टफ्टेड और नॉन टफ्टेड. तो सबसे पहला हमेशा आपको याद रखना है, इफ ए प्लांट बिलोंग टू द पोएसी, रूट के सहित आपको लेना है. ये आपको फील्ड आइडेंटिफिकेशन गाइड में आपको ये ट्रेनिंग दी जाएगी कि जब भी आपका भी ये प्लांट मोनोकॉटिलिडॉन में पोएसी फैमिली को लेना है तो सबसे पहले आप इसके रूट सहित उसको आप निकालो. देन अनदर सिमिलर काइंड ऑफ फैमिली दैट वाज द साइप्रेसी. क्योंकि पहले आप देखोगे तो साइप्रेसी पोएसी अलग नहीं था, दोनों एक ही ग्रुप में था. इन लेटर ऑन इट हैज बीन इज द टू डिस्टिंक्ट फैमिली. तो इस दोनों फैमिली में, जैसे ये दूसरा एग्जांपल मैं आपको दिखा रहा हूं कि जो दूसरी फैमिली है, ये है हमारा साइप्रेसी और ये है हमारा पोएसी. तो दोनों में डिफरेंशिएट करने के लिए सबसे पहले हम देखेंगे इसके स्टेम को. इफ ए प्लांट स्टेम इज द ट्रायंगुलर, आप किसी भी साइप्रेसी प्लांट को आप ले आओ, एवरीवेयर यू विल सी दैट द स्टेम इज द ट्रायंगुलर. इसकी एनाटॉमी, इसका सीधा इसका टीएस काटिए, आप टीएस काटने के बाद इजीली आप देख सकते हो कि ये इसमें जो है हमारा ये करेक्टर्स है अवेलेबल, मींस ट्रायंगुलर. सेकेंडली करेक्टर्स जो हमारा इसमें आएगा साइप्रेसी फैमिली में, दैट इज योर नटलेट्स, इज वेरी इंपॉर्टेंट टू डिस्क्राइब मींस डिफरेंशिएट बिटवीन द जेनरा एंड द स्पीशीज आल्सो. तो इसमें नटलेट्स होता है और दूसरा स्टेम इसका ट्रायंगुलर होता है. इफ आई विल कंपेयर विद दिस फैमिली पोएसी, वेयर यू विल फाइंड कि इसमें स्टेम जो होगा ये सिलिंड्रिकल होगा या टेरेट होगा, और इसमें जो यहाँ पे जो इसका फ्रूट होगा उसको कैरियोप्सिस कहते हैं. दिस इज नॉट कॉल्ड द सिमिलर काइंड ऑफ नटलेट्स. नटलेट्स में कोई कवरिंग नहीं होगा, बट इन केस ऑफ द कैरियोप्सिस आप देखेंगे कि उसमें कवरिंग होता है, सो दैट इज कॉल्ड द कैरियोप्सिस. इन डाइकॉट एक और भी इंटरेस्टिंग है. एक डाइकॉट में आप जब चिनोपोडिएसी और अमरेंथेसी फैमिली के पास जाओगे, वहां पे भी एक सिमिलर कैरेक्टर्स आपको मिलेगा. देयर इज द आर्टिसिल्स, हम उसको बोलते हैं एक कैरेक्टर्स को, जहां पे क्या होता है कि उस नटलेट्स के साथ में ही पूरा कवरिंग जो होता है बहुत चिपक जाता है. तो यहाँ पे वो कैरेक्टर्स नहीं एप्लीकेबल है. हियर द जो इसका बाहर का पेरिकार्प है वो आइदर इट इज द फ्यूज्ड और द इट मे बी द सम ऑफ द टाइम लूजली अरेंज्ड. तो ये दो कैरेक्टर्स के ऊपर में आप पोएसी और साइप्रेसी को इजीली डिस्टिंक्ट कर, मतलब डिफरेंशिएट कर सकते हैं. अब जितना प्लांट आपको अब से अब तक दिखाया, इफ समबडी विल आस्क यू वेदर कैन यू की आउट द ऑल द कलेक्शंस, तो इसमें आपको क्या करना है कि दो ग्रुप आप बनाएंगे. प्लांट हैविंग द पैरेलल वेनेशन एंड द कॉटिलिडॉन्स वन, प्लांट having the reticulate venation and the two cotyledons. इसके बेसिस पे आपने दो ग्रुप बनाया. अब मोनोकॉट में हमने आपको एक प्लांट दिखाया और एक दूसरा प्लांट मैंने आपको एक क्लिंगा दिखाया, दो दिखाया. एक तीसरी और फैमिली हम लोग के पास प्रेजेंट में प्रैक्टिकल है, वो ऑर्किडेसी का, जो अभिषेक सर आपको दिखाएंगे. देन आई विल ओवरऑल व्यू ऑफ द ऑल द प्रैक्टिकल मींस ऑल द मटेरियल्स आई विल डिस्कस. अभिषेक सर. ये सारे प्लांट्स इसको रखेंगे, तो एक साथ उसको दिखाएंगे. कैमरा लेके रखना, ठीक है? और कैसे हम लोग प्रैक्टिकल करते हैं. ठीक है, हाँ ये टेबल पे वो करने के बाद
Hello, dear participants. I am Abhishek Bhattacharjee from Botanical Survey of India. So now I am going to explain you some characters of the family Orchidaceae. Okay, you can see the slides here, and there is a sample plant of orchids. Okay. So in Sikkim circle, where you are there Sikkim, there are a lot of orchids. तो आप ये देख सकते हैं जो ऑर्किड का मेन कैरेक्टर क्या होता है. मैं जस्ट मेन कैरेक्टर आपको बोलूंगा. ऑर्किड्स एक मोनोकॉट्स है, तो इसीलिए उनका जो फ्लोरल पार्ट्स होता है सब 3 या वो मल्टीपल एप्लीकेशन ऑफ 3 में होता है. 3 सेपल होगा, 3 पेटल होगा, सब 3-3 करके होता है. ये इसको ट्राइमेरस बोला जाता है, ट्राइमेरस कंडीशन. ठीक है, आप ये देख लीजिए, ये एक प्लांट है, और उसके बोलने का पहले मैं आपको ऑर्किड के कैरेक्टर बताता हूँ. एक ऑर्किड में जो मेन इंपॉर्टेंट कैरेक्टर होता है उसका जो लेबेलम या लीफ होता है. लेबेलम लीफ क्या है? एक्चुअली दिस इज मॉडिफाइड पेटल. हाँ ठीक है, अभी फोकस आ रहा है थोड़ा. हाँ ठीक है, इससे ज्यादा फोकस शायद नहीं आएगा, कैमरा या इंटरनेट का प्रॉब्लम से ज्यादा फोकसिंग ठीक से नहीं आ रहा है. आई कैन अंडरस्टैंड. हाँ अभी स्लाइड नहीं शेयर कर सकता हूँ क्योंकि वो लॉगिन करने पड़ेगा ना. ठीक है, ओके. तो ये जो ऑर्किड का कैरेक्टर्स होता है, एक मेन इंपॉर्टेंट कैरेक्टर्स होता है. ऑर्किड का लेबेलम या लीफ होता है. लीफ क्या होता है? दैट इज द मॉडिफिकेशन ऑफ द थर्ड पेटल. क्यों मॉडिफाई करता है? क्योंकि ऑर्किड बहुत इंटेलिजेंट प्लांट है, मतलब उसको बोला जाता है जो हाईएस्ट इवॉल्व प्लांट. तो वो क्या करता है, इंसेक्ट को अट्रैक्ट करने के लिए वो उनका जो लेबेलम होता है उसको मॉडिफाई करता है. डिफरेंट कलर उसमें देते हैं, कभी कभी उसमें सैप दे देते हैं. तो इंसेक्ट आके क्या करता है, वो कलर या सैप से अट्रैक्टेड होकर पॉलिनेट करने के लिए आ जाता है. और क्या होता है, गाइनोस्टेमियम और कॉलम मींस आप यू कैन सी इन अदर ग्रुप द स्टेमेन होता है या कार्पेल होता है. ऑर्किड में इंडिविजुअली आपको नहीं मिलेगा. इंडिविजुअल में वन यूनाइटेड स्ट्रक्चर होता है जो गाइनोस्टेमियम या कॉलम बोला जाता है. एंड और क्या होता है, इंफीरियर ओवरी. ऑर्किड का ओवरी इंफीरियर होता है. जैसा आप दूसरे ग्रुप में आप देखे थे सुपीरियर ओवरी है, ऑर्किड में इंफीरियर ओवरी होता है. रिशूफिनेशन क्या होता है, ट्विस्टिंग ऑफ फ्लावर. ये जो आप फ्लावर देख रहे हैं इसमें ये है लेबेलम और लीफ, ये वाला पार्ट. दिस इज द लेबेलम और लीफ. में और थोड़ा सामने लेके आता हूँ. यस, अभी आप देख पा रहे हो, कुछ दिख रहा है. इन दैट केस दिस पार्ट इज कॉल्ड लेबेलम और लीफ. हाँ, ये एक्चुअली इन केस ओरिजिनली अगर ये ऑर्किड नहीं होता तो इसका पोजीशन ऐसा होता, लेकिन ये ऐसा है. व्हाई? बिकॉज ड्यू टू रिशूफिनेशन मींस ट्विस्टिंग ऑफ द फ्लावर. और क्या होता है, पोलेन. इन केस ऑफ ऑर्किड्स पोलेन आर प्रेजेंट इन फॉर्म ऑफ पोलेन मास, दैट इज कॉल्ड पोलिनिया. सो नाउ आई एम गोइंग टू शो यू अनदर photograph. Yes, of this plant, that is Dendrobium ephyllum, the same plant, Dendrobium ephyllum. So here you can see that in Dendrobium ephyllum plant the stem is modified. Here the stem, ये जो stem part है ये modified into pseudobulb. मीन्स ये क्या करता है, स्टोरेज एक ऑर्गन क्रिएट करता है जिसमें वो इन फ्यूचर के लिए, ड्राई सीजन के लिए फूड स्टोर कर सकते हैं. देन ये प्लांट दिखेगा पेंडुलस. ये एपीफाइटिक ऑर्किड है. एपी मींस अबव, फाइटिक मींस प्लांट के ऊपर होता है. एंड ये ड्रूपिंग कंडीशंस में होता है. ये एपीफाइटिक ऑर्किड में, डेंड्रोबियम एफाइलम में, दिस सेपल, द टॉप सेपल इज कॉल्ड एज डोर्सल सेपल. अगर मैं आपको प्लांट फ्लावर दिखाऊं, दिस इज द डोर्सल सेपल. and this part is the petal. This is the petal. ये पिक्चर में आप देखिए, this part is the petal and this is the lateral sepal. ये जो साइड में आप जो सेपल देख रहे हैं ये है lateral sepal. पिक्चर में आप देखिए this is the lateral sepal and this structure is labellum or lip. ये वाला जो पार्ट है ये है labellum और lip and इसका अंदर और भी माइन्यूट पार्ट है जैसे कॉलम, पोलिनिया, वो आप नेकेड आई से इधर से आप नहीं देख पाएंगे, मतलब ऑनलाइन में वो नहीं होता है, वो आपको ऑफलाइन क्लास जब होगा या माइक्रोस्कोप में, डिसेक्टिंग माइक्रोस्कोप में आप देख पाते हैं. तो दिस स्ट्रक्चर आर द पोलिनिया मींस द पोलन मास. देखिए आप पोलन इन द फॉर्म ऑफ मास ऑर्किड में होता है and the other character is the column, that is the gynostemium. So these are the main character of orchids and apart from these characters the other character is that orchids में seed जो होता है वो बहुत minute dust like होता है, क्योंकि ये seed क्या करता है, ऑर्किड एपीफाइट भी हो सकता है, टेरिस्टियल भी हो सकता है, ये डस्ट लाइक सीड है. इसका मतलब वो सीड बहुत दूर दूर एयर बोर्न होके जाके वो जर्मिनेट करते हैं. लेकिन इधर एक क्वेश्चन है, ऑर्किड में मिलियंस ऑफ सीड्स होता है, अगर सब सीड्स जर्मिनेट कर जाता तो पूरा सब जगह में आपको ऑर्किड ही ऑर्किड दिखता है, लेकिन ऐसा तो एक्चुअली में नहीं होता है ना. ऑर्किड तो ज्यादा नहीं मिलते हैं, आपका हिल एरिया में ज्यादा है, लेकिन हम लोग को इधर तो ऑर्किड मिलता ही नहीं है. तो व्हाई? बिकॉज ऑर्किड सीड जहां पे गिरेगा वहां वो जर्मिनेट नहीं करेगा जब तक ना एक टिपिकल माइकोराइजा उधर रहेगा. सो ऑर्किड सीड्स, जर्मिनेशन ऑफ ऑर्किड सीड्स इज डिपेंडेंट ऑन टोटली माइकोराइजा प्रेजेंस. तो माइकोराइजा वो सीड को एक्चुअली उसका जो सीड होता है वो एक सेल्बुमिनस सीड होता है, क्योंकि वो सीड का अंदर कुछ खाना नहीं रहता है, तो इसलिए सीड को सरवाइव करने के लिए माइकोराइजा का हेल्प चाहिए होता है. तो इसीलिए ऑर्किड सीड्स, जर्मिनेशन ऑफ द ऑर्किड सीड्स इज टोटली डिपेंड ऑन माइकोराइजा. सो दीज आर द मेन कैरेक्टर्स ऑफ ऑर्किड. थैंक यू. नाउ आई एम रिक्वेस्टिंग डॉक्टर गोपाल कृष्णा टू जॉइन.
कैमरा करोगे ना?
हाँ, उसको बताएंगे ना कैसे करना है.
कैमरा में बता इधर भी ठीक था.
Say for example, एक हमारा collection bag है and we having these all collections and how to segregate the plants in the field, because अगर आप segregation नहीं कर सकते हो तो आपको सबसे बड़ी difficulty identification में होगी कि कैसे हम इस plant को identify करेंगे, और उसमें आपका time बहुत जाता है. तो सबसे पहले आपको क्या करना है, यहाँ पे जो हम classify करेंगे simply based on the characters. जैसे अभी हम क्या देखेंगे कि monocot और dicot का plant हम कैसे उसको differentiate करते हैं. तो ये monocot orchidaceae, that's okay. Another plant that
another find monocot.
No, where it is? What is the monocot?
एक ऑर्किड शो कर दिया ना.
हाँ, that's okay. The another is the dicot. ये हमने इसको भी dicot कर दिया, ये सारे स्पेसिमेन को हमने dicot कलर कर दिया.
It's okay. Then you should
Again, you see this, the parallel venation and this monocot. इसको हम फिर से अलग कर दिए. ये मेरा monocot है, इसको भी मैंने अलग कर दिया.
ठीक है, combination.
This is the monocot. This is the poaceae and cyperaceae. This is also the monocot. ये भी हमारा monocot कर दिया. अब ये सारे specimens हमारे एक साइड में dicot हो गए और एक साइड हमारे monocot हो गए. Just I'm showing that how we are making the key out. ये फील्ड में हम ऐसे की आउट कर रहे हैं, आप अपने कॉपी में भी ऐसे की आउट कर सकते हो. हाउ टू मेक ए की. ये बनाने के बाद अब इसमें से फॉर एग्जांपल विद इन द मोनोकॉट, इस मोनोकॉट में देखिए एक प्लांट का क्या है कि स्टेम काफी वुडी है. सो स्टेम वुडी वन ग्रुप,
अनदर ग्रुप जो हमारा पास में हो गया यहाँ, द स्टेम वेरी हर्बेशियस कह सकते हो, या यू मे से दैट वेरी सॉफ्ट और डेलिकेट। तो ये हमारा एक दूसरा ग्रुप हो गया। तो मोनोकॉट के अंदर ही हमारा दो ग्रुप बन गया, वन हैविंग द स्टेम इज द बॉडी, और दूसरा ग्रुप का जो है वो हमारा सॉफ्ट स्टेम है। अगेन अब इसमें क्या देखेंगे, फ्लावर्स लार्ज एंड सोई। इन द केस ऑफ द कमेलिनेसी आप देखेंगे ये फ्लावर्स काफी सोई होता है और बड़ा होता है। आप इसको देखेंगे, ये प्लांट हमने फिर अलग कर दिया, हमारे पास क्या बच गया, ये दो प्लांट्स एक साइड आ गए, एक साइड हो गए, तीन ग्रुप हमारे बने। मोनोकॉट के अंदर में हमने तीन ग्रुप बनाया, जिसमें फ्लावर लार्ज है। अगर आप ये जानते हो फैमिली का नाम डायरेक्ट कर सकते हो कि दिस बिलोंग्स टू द कमेलिनेसी। अब हमारे पास इन दोनों के पास में थोड़ी कंफ्यूजन हो गई, बोथ लुक्स वेरी सिमिलर, ये दोनों प्लांट हमारा सिमिलर लुक लग रहा है। अब इसमें थोड़ा सा आपको कंफ्यूजन हो रहा है, तो हम क्या कैरेक्टर्स इसमें अप्लाई करेंगे। ये देखिए इन्फ्लोरेसेंस सीधा देखिए दोनों में डिस्टिंक्ट है। ये हियर द इन्फ्लोरेसेंस इज द पेनिकल, और यहाँ पे क्या देखोगे एक हेड में है, और इसके साथ में जो ये ब्रैक्स जैसा स्ट्रक्चर्स है ये इसके साथ में लार्ज है। सिंपली लिखो इन्फ्लोरेसेंस इन हेड एंड इन्फ्लोरेसेंस इन द पेनिकल्स। स्टेम इज द ट्रायंगुलर और स्टेम इज द सिलिंड्रिक। हमने बना दिया साइप्रेसी, पोएसी, कमेलिनेसी एंड दिस ऑर्किडेसी। ये हम बेसिक स्टैंडर्ड तक की आपको बताया कि कैसे हम की आउट करते हैं, लेकिन इफ यू आर वेरी इन एडवांस स्टेज then you have to apply some more morphological character. Say for example, ऑर्किडेसी फैमिली में पॉलिनिया होता है, उसमें आप इस बार बाकी तीनों फैमिली में नहीं होता है, यू कैन अप्लाई ऑर्किडेसी। यूजुअली एपिफिटिक एंड द फेरिस्टियल दोनों होते हैं, बट ये सारे जो प्लांट होते हैं कोई भी एपिफिटिक नहीं होता है। तो ये अपने ऊपर में है कि आप कितना करेक्टर्स आप ऐड कर सकते हो, और ऐसे करेक्टर को ऐड करो कि जो इसमें है पर इसमें नहीं है। कंट्रास्ट करेक्टर जिसको बोलते हैं, दैट इज वेरी इंपॉर्टेंट इन आइडेंटिफिकेशन। अगर आप ये टेक्निक सीख लोगे तो लाइक वाइज यू कैन अप्लाई हियर आल्सो। अब हम डाइकॉट में बताते हैं कैसे कर सकते हैं। टू ग्रुप प्लांट मैंने आपको बताया था, पॉलीपेटली, गैमोपेटली और मोनोक्लेमाइडी। तो सबसे पहले हम इसको पॉलीपेटली में करते हैं। हियर पेटल्स आर फ्री, सो पॉलीपेटली। हियर पेटल्स आर फ्री, so this will be go to the polypetally. Here this is the monoclemidy because here variant है, यहाँ पे एक होल में है, so that is why हम इसको यहाँ पे monoclemidy में रखेंगे। Again solanaceae, this is the gamopetally because petals are fused. Legumes, this will be going to the polypetally. Myrtaceae, this will be going to the polypetally. Aristolochiaceae, this will be going to the monoclemidy. Brassicaceae, polypetally. Rhytia, gamopetally. Legumes, polypetally. Artocarpus, monoclemidy. Rhytia, this will be into the gamopetally. Ficus in monoclemidy. Morinda, this will be going to the gamopetally. This is the Scopodia, this will be into the gamopetally. Then Rolkia, this will be also going to the gamopetally. This will be going to the gamopetally. This Acaranthus will be going to the monoclemidy. Solanum, this will be going to the gamopetally. This is the Cucurbitaceae. This will be the polypetally member, 75 number family. Yeah, right. This is the Ficus, this will be going to the monoclemidy. This is your Morinda. Already is on. Okay, leave it. Morinda will be in the gamopetally. Solanum diphyllum, this will be going to the gamopetally. Then your this is the Alpinia, this will be in the monocot already. Sorry, right. So this is the Padolgia, this will be going to the monoclemidy. Monoclemidy. This is the Gymnema. This will be Tylophora. This will be going to the gamopetally. अभी देखिए मैं बाकी और भी सारे प्लांट है, मैं टाइम के शॉर्टेज की वजह से मैं आपको बता रहा हूँ कि कैसे हमने किया। This is not the one day job that I am telling you that this is the poly, this is the gamo, this is the monoclemidy. This is the experience what I am doing. पर for the beginners I am telling, आपको फ्लावर्स देख के करना है। ऐसा नहीं कि हमने बताया अच्छा उन्होंने ऐसा ऐसा बताया तो आप भी कर लो। आपको फ्लावर देख के करना है। Since I have memorized all these things, so that is why it is easier for me. But for you, you have to see the corolla or the flower characters first. Remember, पॉलीपेटली, गैमोपेटली और मोनोक्लेमाइडी। अभी आप क्या करना है, जब ये 3 फैमिली हमने कर दिया, अब इसमें भी हम की आउट कर सकते हैं। कैसे की आउट कर सकते हैं वो मैं आपको बता रहा हूँ। से फॉर एग्जाम्पल दिस इज अ गैमोपेटली, सबसे बड़ा ग्रुप गैमोपेटली में ज्यादा स्पीसीज आते हैं। इसमें हम कैसे ग्रुपिंग कर सकते हैं, प्लांट क्लाइंबिंग और क्लाइंबिंग श्रब्स और क्लाइंबिंग हर्ब्स और ट्विनर, सारे क्लाइंबर्स को हम एक साथ बाहर कर देंगे। अगर हम इस ग्रुप की बात करें, दैट इज द सिंगल क्लाइंबिंग प्लांट्स, तो हम इसको यहाँ पे बाहर कर देंगे। और प्लांट हैविंग द व्हाइट लेटेक्स, ये देखिए, ये इसमें व्हाइट लेटेक्स होता है, इसमें व्हाइट लेटेक्स होता है, इसमें व्हाइट लेटेक्स होता है, ये सब को हम इसमें बाहर कर देंगे। और ये पर्टिकुलर फैमिली में बिलोंग करता है। एट प्रेजेंट दीज आर बिलोंगिंग टू द अपोसाइनेसी। प्लांट हैविंग नो वाटर व्हाइट लेटेक्स, ये सब इधर हम बाहर कर दिया। व्हाइट लेटेक्स की जैसे ही बात किए हमने, इसमें ये सारे प्लांट को ले आया और हमारा एक पर्टिकुलर फैमिली है अपोसाइनेसी या स्क्लेरोपेडेसी को शो करता है। फिर इसमें आप अप्लाई करो ट्री स्पीसीज, हर्ब स्पीसीज और सर्ब स्पीसीज, तो ये एक और कैटेगरी आप बना सकते हो। इसमें जितनी भी ट्री होगी वो सारे हमारे इधर से बाहर हो जाएंगे। मोरिंगा बाहर हो गया, राइटिया हमारा बाहर हो गया, और श्रबी एलिमेंट हमारा इधर सब आ गया एक साइड में। तो श्रबी एलिमेंट 2 तरह के हो सकते हैं, आइदर इट मे बी द क्लाइंबिंग और इट मे बी द इरेक्ट, दिस ऑल डिपेंड्स अपॉन। तो इसको हम यहाँ पे जाकर के हम इसको बाहर कर देंगे। जब आप ये सारा कैरेक्टर्स को, ये हमारा कैरेक्टर्स हो गया, एक ट्री स्पीसीज हमारा यहाँ पे तैयार हो गया। अब ये ट्री स्पीसीज को हम फिर से, इसमें हमारे पास 2 स्पीसीज आ गई ट्री स्पीसीज। एक ट्री स्पीसीज में क्या है कि व्हाइट लेटेक्स आ रहा है और दूसरे में हमारे पास में कोई व्हाइट लेटेक्स नहीं आ रहा है, फर्स्ट थिंग। सेकेंडली, इस ट्री के बार्क पे देखोगे छोटे छोटे लेंटिसेल्स हैं, बट इन दिस केस देयर इज नो लेंटिसेल्स। लेंटिसेल्स इज नथिंग, इट इज ए ब्रीदिंग पोर्स, यू मे से दैट। तो ये वाला जो कैरेक्टरिस्टिक्स अगर आ गया, और दूसरा और भी लगा सकते हो, हियर लीव्स आर नॉट सिंपल, दीज आर द कंपाउंड लीव्स, और इसमें आप देखेंगे लीव्स आर सिंपल। तो कैरेक्टर्स हमारे सामने रखा हुआ है, सिंपली यू हैव टू अप्लाई द डिस्क्रिप्शंस। जो मैंने आपको पहले बताया था कि जो भी कैरेक्टर्स आपको इस प्लांट में दिखेगा वो सारे को आपको मॉर्फोलॉजिकल डिस्क्रिप्शंस के रूप में लेके आना है। Once you are ready to do this, I think you are the almost close to the identification of the plant. So I think this is sufficient, and because of the time, I'm stopping my talk here. If you have any query, you are welcome to ask the question. Over to Basant sir.
बसंत कहाँ गए? कितना टाइम ले लिया हमने? 2-3 घंटा था।
Over to Basant sir.
Over to Basant. Na Basant na এখন over Google sir.
Okay. Over to you sir.
Now it is my opportunity to introduce with the team of CNH who participated and who demonstrated about different families. So Dr. Anand Kumar, जाओ. हाँ कैमरा तो ऊपर करो.
उससे ऊपर करो.
हाँ हमको दिखने रहे. तो ये है डॉक्टर अनंत कुमार, डॉक्टर गोपाल कृष्णा.
Thank you, Gopal Bhaiya. Is there any question from the audience?
Dr. Shyam Biswa.
Is there any question?
And
because now we will move to the valedictory session. So before that, if there is any discussion or question, you can ask.
And myself, Abhishek. Okay, then over to.
Yes.
Yeah.
Already moving forward.
Now,
Yes, I know.
The team of CNH who participated and who demonstrated about different families. So, Dr. Anand Kumar. Camera So,
Can we move to the validity session? Gopal Bhaiya, no more question. No question from the audience. कोई सवाल नहीं है गोपाल भैया.
Okay, thank you. We are happy that you guys know question. Otherwise, I should have asked earlier.
So can I have a question?
Yes.
This is Dr. Sanjeev Borwa from Brulin University, Kokrajhar. This is actually not a question. First of all, I'd like to see all of you together. Okay, and my question is there any planning to BSI organize a workshop on nomenclature in the upcoming days?
Abhishek is the right person to respond to this.
Yes.
Actually, we are organizing nomenclature course several times in different part of the countries. So in future also we are having some plan, but not means actually in local in Kolkata or in adjoining areas we have already conducted. Last month we conducted in Rahul Ramakrishna Mission. So in upcoming days, particular on nomenclature, we don't have any plan. But maybe in future we can means organize, and in that case our directors are at headquarters. If they instruct from there, and if some other institute request them, in that case we can organize.
Right, sir. So thank you so much, sir.
Okay.
With us from YouTube Live. The passcode of program is on your screen. Please note down that passcode to submit your feedback and simultaneously to download your e-certificates. Code will be visible on your screen. Kindly note down. The passcode is SHRC 2022.